Right. Go live. How do I go live? Right. Hello, everyone. Everyone can see and hear me, but I can't see my screen. All right. Welcome to this conference. I had some slides prepared, but not quite sure that. Okay, so start again. Welcome to the student conference, the PhD student conference in 2021 after last year's was cancelled. It's a virtual conference this time, hosted by DMX, and they have put a lot of work into preparing us all. It has been a very, very unusual year with the announcement of the pandemic last year in March. Schools closed, institutions closed, and many countries went into lockdown, installed curfews, and we all basically had to go and work at home while also looking after children. Fieldwork was cancelled. We couldn't conduct interviews or indeed access the lab, and that has put a lot of strain on all of us, but particularly you, and the concerns about how you can continue with your projects. Many of you went and did writing of your thesis or parts of result chapter, analyze data, or learn new skills. But it wasn't just writing that obviously concerned you. Many had to look after or were concerned about children, family, and friends, about their health, but also how to look after each other while continuing with your work. So it has been a very difficult year. We very quickly learned to use all sorts of technology to stay in contact with each other, or stay in contact with supervisors through email, Skype, WhatsApp, Teams, Zoom, and other methods. And here we are again doing exactly the same. The student conference last year was cancelled because obviously with the pandemic, it wasn't useful. It didn't make any sense to continue meeting in person. But despite all of that and all the difficulties that you and everyone else has faced, you continued with your work. And last year, 18 students submitted their thesis and 22 students had their viva. Again, all of them on Zoom or through other technology, which is not always easy. And 16 students have completed all the way through, which is a fantastic achievement given the circumstances. Another thing that has really impressed me over the last year is those who could and had the time, because they didn't have to shield or look after others, the extraordinary response that there was in supporting local communities, supporting hospitals and health facilities, or going back into the lab to work on the COVID response. A lot of you have worked, taken out time of your studies and worked in all sorts of ways in which you could help and support communities and patients, and that is it's extraordinary for as a community how much you have achieved over the last year in not just looking out for yourself, but actually looking out for others and giving what you possibly could give. Another aspect that I thought was just really fantastic is over the last year, I've read basically all of your pub reports. And what really struck me is that despite the difficulties in adapting your projects or not being able to do what you plan to do, how positive many of the reports were, and who spoke about the extraordinary progress you have made, the flexibility you've shown and adapted, together with your supervisors, your projects, found other ways to address a question, and often followed that alternative question or changed topic with enthusiasm despite all the challenges you've faced. And that has made me really, really proud of how resilient you have been and continue to be, because the pandemic is really not over in anywhere in the world. So I think as a community, you can be really proud of yourself and what you have achieved over the last year, despite everything that has been going on. And so without any further ado, I would like to start and welcome you to the conference. It is obviously slightly different this year. We are not meeting face-to-face. We are meeting in a virtual environment, which makes it, which loses some aspects of what we enjoy, talking to each other, with all the complexity that you can't get on screen. On the other hand, I think it's an enormous opportunity to actually network and talk and see the work of all students across the globe. And we have about 50% of our students are not based at LSTM or in Liverpool. They're based in other places. And the way this conference works will make it easier to network and chat in the different outbreak areas and poster booths, etc., with colleagues from across the globe. What I really would like from you, both supervisors and students, is after the conference that you provide us with feedback, how that worked and what you thought worked well and how you think we can incorporate that in the future. Because I think maybe the only good thing that came out of the pandemic is that we are learning to use technology much more efficiently to connect, basically almost without borders or without the need for aeroplanes with each other. So please provide us with feedback at the end and ideas how we can incorporate that in the future. Now I have to do the usual household announcements. If the fire alarm goes off, that is obviously in your house and not in a conference facility. So please visit the expo area where you can connect with other students and their work. You need to register your interest with a student, for instance, to discuss the posters. We've got quite a few networking and breakout sessions in the afternoon, I think, across a wide variety of topics. So please join these sessions. I think there's something of interest for everyone, and also the networking will allow you to connect with other people. So I hope you enjoy the event. I certainly look forward to it and learn more about the work you all have done. Thank you.
Okay, hello and welcome. So I'm Eric Lucas, and it's my pleasure to introduce the first session of talks for this conference, which I have to say I'm really looking forward to after reading the abstracts. So we'll kick off with Melanie Nolden, who'll be talking about the influence of cytochrome B5 on the expression of some important resistance-associated cytochrome P450s.
Okay, thank you, everyone, and hello. I'm Melanie, and I'm a third-year PhD student of the Vector Biology Department of LSTM, and I'm mainly funded by Bayer Crop Science in Germany and also partly funded by the IVCC. And within the next 10 minutes, I'm going to talk about the influence of cytochrome B5 on heterologously expressed CYP6P9A and B. So first of all, cytochrome P450 monooxygenases, or CYPs or P450s, are generally involved in the phase 1 metabolism in almost every organism. And in pests or mosquitoes, special P450s are responsible for the pyrethroid metabolism. And pyrethroids are heavily used in vector control interventions. And genetic or biochemical changes within these enzymes then can lead to a faster or better metabolism of pyrethroids. And then we have the so-called metabolic resistance. So in general, if we look at this slide, this is the metabolism of P450 enzymes. They are substituting their respective substrate with an extra oxygen here. And for that, the input of 2 electrons is necessary. And P450 enzymes are incorporated into the membrane and in the endoplasmic reticulum. And also, we see here this enzyme, the cytochrome P450 reductase. This enzyme is responsible for the transfer of the 2 electrons from the electron donor NADPH. And then we also have a third party incorporated into this membrane fraction, and this is the cytochrome B5. And this protein is probably responsible for the faster input of the second electron, for the alteration of the CYP conformation so that the electron acceptance is facilitated, or therefore that the CYP and the B5 are building a complex, and so we have an accelerated reaction of receiving the second electron, or cytochrome B5 is stabilizing the enzyme. So within the last years, a lot of CYPs had been recombinantly expressed, also CYP6P9A and B of the malaria mosquito Anopheles funestus. However, to date, only the E. coli expression system was used, and in my approach, I'm using the baculovirus expression system with a subsequent high 5-cell expression. And also to date, only Anopheles gambiae CPR was used in the co-expression, and the cytochrome B5 was only added to the reaction mix, and also only Anopheles gambiae B5 was used. And within the next minutes, I'm showing you my results where I also used the Anopheles funestus CPR, Anopheles funestus B5, and used the co-expression. First of all, I wanted to know how is the expression level of the CPR in the cytochrome B5 in Anopheles funestus. So we are rearing the Fumos R strain, which is pyrethroid resistant, and the susceptible Fung strain. So I isolated RNA, did the cDNA, and conducted qPCR analysis. And here you see at the starting point the expression levels of CYP6P9A and B in both strains, and especially the B variant is massively upregulated in the Fumos R strain if you compare it to the Fung strain. However, if we have a look at the expression level of cytochrome B5 and the reductase, we see that we didn't observe any significant differences at all. And this is very interesting, especially if we look at this fold change for the B variant here. So after I expressed my enzymes in the baculovirus expression system and the high 5 cells, you're containing your cells, and they have the endoplasmic reticulum with the P450 enzymes, these little Pac-Mans here. And then you isolate this microsomal fraction by several centrifugation steps. And afterwards, you resuspend them in special buffers and incubate it with model substrates. And in our lab, we are using mainly coumarines. And as well as I said at the beginning, NADPH as an electron donor is vital for the reaction. In my case, I was using benzyloxymethoxytrifluorocoumarine, BOMFC, and this is looking like that. And what is happening if we expose this coumarine to P450 enzymes or microsomes? They are cutting this molecule here, and then we're receiving this molecule, which is luckily fluorescently active. So in the end, we can then measure the fluorescent activity and then associate this with the metabolic activity. And as I said in my introduction, I've done this with the Anopheles gambiae CPR. And here on the left, you see the results for the A variant of the enzyme. And here you see it for the B variant. And for the Anopheles gambiae CPR, I received activity around 150 picomole product per minute per milligram protein, and it's the same for the other enzyme. And these are the results for the Anopheles funestus CPR, and I received a similar pattern. It's slightly different, however, I wouldn't say that it has a massive impact. So now we know we can use, at least for these enzymes, both reductases. Then I did for the first time the co-expression of Anopheles funestus cytochrome B5, and as you can see, the metabolic or fluorescent activity is enhanced, and also here for the B variant of the enzyme, it's almost doubled. And then as well, because I wanted also to check what was done in the past, I've added human cytochrome B5 to the reaction mix. Because this is commercially available. And we also see that we have a massive increase in the fluorescent activity. But what does that mean now with regards to the actual pyrethroid metabolism? So then I've conducted LCMS analysis with CYP6P9A and also later UCB. In red, you see the enzyme with co-expressed B5 and in black without. And on the first graph here, the delta mesrin depletion. And you see that over time, delta mesrin is depleted. So we see a decreased concentration of delta mesrin. And also we see slightly significant differences between co-expressed B5 and without. However, I wouldn't say that it's massive. And here you see the respective 4-hydroxy delta mesrin. This is the first metabolite of the delta mesrin metabolism, and we see that the formation is increasing, and then we have a plateau here, and we didn't observe significant differences if expressed with or without B5. For the B variant of the enzyme, it's actually looking completely the same, the patterns, and we didn't observe differences in delta mesrin depletion if B5 is co-expressed with the enzyme. So in summary, I can say that we didn't observe significant differences of cytochrome B5 and the CPR expression levels between the susceptible fung and the resistant fumose R strain. We didn't observe big differences in the fluorescent probe assay between the Anopheles gambiae and the Anopheles funestus CPR, so we can now say that both of them can be used in this approach. Also, adding human cytochrome B5 and the B5 co-expression are enhancing the metabolic activity towards BOM-FC, or maybe also other coumarin substrates, however, not towards delta mesrin. So as a conclusion, I can say that cytochrome B5 is not necessary for recombinantly expressed CYP6P9. However, it can enhance the reactions. And with that, I would like to thank Ralf Naun and Mark Payne, Sebastian Horstmann, Sarah Rees, and Uwe Pluschgel, the Molecular Entomology and Toxicology Lab, Bioenvironmental Science, and especially the insect rearing team, and the IVCC. So thank you very much.
Okay, thank you, Melanie. That was a really great talk. If people have questions, by the way, please do write them into the chat. In the meantime, Melanie, can I ask, why do you think human B5 seemed to work better than Anopheles funestus B5? Is it just because you were adding it physically, the sort of ready-made B5, so that there was more of it, or could it be something else?
Unfortunately, I haven't measured yet the concentration of B5 in my co-expressed approach. So for now, I would say that the concentration of B5, which I was adding, is just higher in the co-expressed approach. Does that make sense?
Yeah, sure. And so in the delta mesrin experiment, maybe you said this, and I apologize if you did. was that with the added B5 or was that with the co-expressed B5?
Co-expressed B5.
Okay,
Yes.
so did you try, is it possible that if you added B5, could it be that there would be a stronger effect, do you think? Is it just a increased quantity of B5 that's necessary?
As I have seen that also in the fluorescent assay, the activity was doubled with my co-expressed B5. And in the delta mesrin experiment, I didn't observe, massive or actually no difference. I wouldn't say that adding B5 would make such a difference.
Okay, cool. Thank you. And there's a quick question from the audience as well then. An interesting finding, how similar are the sequences of B5 in different anopheles? Do you have any idea why B5 is important for some P450s but not others?
So I think these are two questions, right? So we have very conserved parts in B5 throughout Diptera et al. And also I would say every organism. And what was the second part?
Do you have any idea
is it
why
important?
Yeah.
Okay. No, I haven't had a look into that. However, we have quite a few P450s expressed in our lab, and I would really be interested in to have a look how this is affecting. We have tried with some of B P450, and we didn't observe also massive difference with and without B5 so far.
Cool. Thank you very much, Melanie. That was great. Very interesting. Thank you. So please do type your questions in the chat. Feel free to type your questions in the chat as the talks go along, by the way, and then we can we'll have a set of questions ready at the end of the talks. So next up we have Nesrin Alomran, who will be telling us about experimental antivenoms for hemotoxic snakebite.
Thank you, everyone. Good morning, everyone. And I will talk about pathology-specific experimental antivenoms for hemotoxic snakebite, the impact of immunogen diversity on in vitro cross-reactivity and in vivo neutralization. So snakebite envenoming is a neglected tropical disease with high rates of morbidity and mortality annually. Most snake venoms can be broadly classified according to the main pathophysiological effects that caused, such as cytotoxicity, neurotoxicity, or hemotoxicity. Hemotoxicity is one of the most common critical signs in snakebite victims, particularly by viperine snake and some other snake species. Coagulopathy toxins are typically serine proteases or metalloprotease, and these toxins can cause venom-induced consumption coagulopathy by activating different blood clotting factors or actively depleting fibrinogen. Snake venom proteins responsible for causing blood coagulation disorders can be categorized by either anticoagulant or procoagulants according to prolonged or shortening blood clotting cascade. Antivenom is the only treatment for snakebite with some limitations such as poor safety profile, low specificity, having geographically limited efficacies, and of course the financial cost. So the rationale for this study is, can pathology-specific antivenoms have a global efficacy? To identify the potential pathology-specific antivenom by using two different sets of immunogens to raise ovine polyclonal antibodies against hemotoxic snake venoms, to assess the binding and neutralizing capability to assess the binding and neutralizing capability of resulting experimental antivenom against snake venom immunogen. To do so, we're generating ovine polyclonal antibodies against two groups of venom immunogen, which contain 7 coagulopathic venoms and 12 hemotoxic venoms. Both containing venoms source from wide geographical distribution, and we then assess the specificity and inhibitory capabilities of the two resulting experimental antivenom by using in vitro immunological and venom neutralization assays before testing their preclinical efficacy. So here the SDS page gel for the venom profile for all individual and mixture venoms, and the immunoblot results reveal that both experimental antivenoms were able to detect the vast majority of the proteins found in snake venom toxins. And we then did the endpoint titration, ELISA, to quantify the levels of binding between sheep sera or purified IgG and the venom immunogens. The result of the analysis with the two experimental antivenoms revealed extensive and comparable binding level to each of the different immunizing mixture. And the most noticeable to this was the cross-reactivity with the venom from Callocelisma rhizostoma, which exhibit lower binding level with both experimental antivenoms. And we then did the avidity ELISA to determine the strength binding between antibody and antigens in the presence of an increasing concentration of cotropic agent. And the results revealed that both experimental antivenoms exhibit highly comparable and potent binding to both mixtures and individual venoms, with a slight decrease in binding level were observed for both experimental antivenoms, with maximum concentration of cotropic agent at 4 molar. Similar to the endpoint titration ELISA, there were no major differences between binding to the different venom except in Callocelisma rhizostoma, which has weaker binding compared to the other immunogen. In comparison, semi-polyvalent, the positive control show much weaker binding against both venom mixtures and individual venom. And we also did the chromogenic assay serine proteases activity, which is a kinetic enzymatic chromogenic biochemical assay to quantitate the snake venom serine proteases activity and their inhibition by both experimental antivenoms. Of the 12 venoms, only 7 venoms exhibited venom activity compared to the negative and positive control. And experimental antivenom 2 exhibiting over 50% reduction against 4 of 7 venoms, whereas experimental antivenom 1 exhibited high level of snake venom serine proteases inhibition against beta-serotins and Trimeresurus alpolopris, which is a highly surprising result since neither these venoms used as venom immunogen to generate experimental antivenom 1. And we did the snake venom metalloproteases assay, which is a fluorescent enzymatic biochemical assay to quantitate the snake venom metalloproteases activity and their inhibition by both experimental antivenom. Of the 12 venoms, only 3 did not show any activity. In general, between 40 to 80% of the activity was reduced by experimental antivenoms, and comparison between both experimental antivenoms revealed highly similar result of 4 of 12 venoms tested. And then we did a plasma coagulation assay to assess the neutralization of and the clotting activity of coagulopathic venom toxins. And of 12 venoms, 10 exhibited procoagulant activities as evidence of increase in kinetic profile compared to the negative control, which was plasma only. The other 2 venoms were excluded from the study. Both experimental antivenoms exhibited anticoagulant activity compared to the negative control. In general, both experimental antivenoms outperformed the commercial antivenom semi-polyvalent in all enzymatic assay. And finally, we did murine preclinical efficacy experiment to assess both experimental antivenoms efficacy against hemotoxic venoms. So 8 venoms were selected, 4 that were included in both experimental antivenoms, and 2 that were only included in experimental antivenom 2, and the last 2 were not used in either immunogen mixture. Both experimental antivenom exhibited limited protection against callosal smarodestoma, which correlates with our earlier observation of reduced immunological cross-reactivity. And both experimental antivenoms failed to provide any protection against Leishmaniasis muta, while both experimental antivenoms exhibited potent paraspecific preclinical efficacies against Vibrio amidate. To conclude, our finding revealed that careful selection of specific venom or toxins used as immunogens is a fundamental importance for stimulating neutralization against diverse snake species. Our in vivo studies demonstrated that adding additional venoms to the immunogen mixture to increase paraspecific efficacy could potentially counterproductive by diluting these toxins. Diluting these antibodies specific to the toxin, leading to the decreased cross-reactivity or neutralization as in experimental antivenom 2. There are a number of limitations with this study. For example, only a single animal was immunized with a single dose to generate experimental antivenom due to both ethical and financial constraints. Despite these limitations, our findings highlight at least some evidence that generating anti-hemotoxicity antivenoms with global efficacy may be a viable future strategy for tackling snakebite envenoming. At the end, I would like to thank my colleagues who involved in this study, and special thanks to my supervisor, Professor Nicholas Caswell. Thank you.
Thank you very much, Nesrin. Another really great talk. It's really interesting. So I'm not at all an expert in this field. So one question I have. So if I understand correctly, what you did was you developed antibodies by exposure to sort of all of these toxins simultaneously. What would an alternative approach be to develop all of these antibodies separately by exposure to each venom separately and then just mix the antibodies after that? Or am I being really naive, and is there a really good reason why that's not an option?
We make a mixtures of venoms and then we mix it into the sheep to generate antibodies. But
so would an alternative be to put each venom individually, independently into different sheep and get a single and get one antibody against each venom and then mix the antibodies? So instead of mixing the venoms, you mix the antibodies. Is there a reason why that?
No, there is no reason, but we need to make polyspecific antivenoms against the hemotoxic snake venom.
Okay. Yeah. Cool. Thank you very much. Really great talk. Really interesting. Thank you. So due to technical difficulties, we unfortunately can't have our scheduled third speaker, Sean Tomlinson, in today. So instead, Salma Belnour has kindly agreed to step in with her talk. It's a pre-recorded talk, but Salma will be there to answer your questions at the end. So do pop your questions into the Q&A tab in the chat. So here then is Salma's talk on fecal volatile organic compounds.
Hello, everyone. My name is Salma. I'm PhD student year 4. Today, I will talk about our research on fecal volatile organic compounds in pediatric inflammatory bowel disease. Inflammatory bowel disease is a lifelong relapsing condition of unknown etiology. So novel approaches are needed to better understand the underlying mechanism of the disease. More recently, the interest has been growing in the role of the focus in diagnosing and monitoring of many diseases. So what are FOX? Volatile organic compounds are small organic molecules which are easily evaporate at room temperature. They result from the metabolism of intestinal mucosa and gut microbiota. FOX emitted from stool are responsible for its smell. Therefore, characterization of fecal focus in intestinal disease may provide a new insight into underlying disease mechanism. Objectives. We tested whether focus in stool samples different in IBD cases with those with other gastrointestinal disease. we evaluated if fecal focus differed between Crohn's disease, ulcerative colitis, and IBD unclassified. We evaluated if these fecal focus in children with IBD changed after 3 months of treatment. This study was a prospective observational study of children who suspected IBD and attending 3 specialist clinics in the UK. We got stool samples from these children collected at home and sent to the hospital and stored at -80. Diagnosing of IBD was confirming by usual clinical procedures. Then IBD cases were matched with non-IBD controls by age, sex, and site. Focus were determined by gas chromatography mass spectrometry or GCMS. Raw data were processed using a standard analytical method. The statistical analysis was performed by t-test and INOVA in addition to supervised classification analysis and Perminova. Results, we recruited 132 IBD cases and controls. As you can see, this table shows demographic variables, diet, and whether they use antibiotic were similar across 2 groups. This slide shows the differences between fecal focus in IBD cases and controls at baseline. The diagram on the left is SPLS plot, and it shows the analysis that uses all of the Fox data and best separate the IBD case children, which is shown in blue triangle, from control children, which is shown in red circle. This diagram is 2-dimensional presentation of a 3-dimensional data, so it is actually underestimate the separation between 2 groups. The loading score on the right shows the abundance of 16 Fox were lower in IBD cases. The compound going on the left of the diagram were lower in IBD cases, and these mainly carboxylic acid and short-chain fatty acid, ketones, and aldehyde. 2 compounds which is going to the right of the diagram were statistical significance higher in IBD cases, and these phenol and propan-1-ol. The statistical model used to generate these data has an error rate about 30%, which shows the model performing well with these data. This slide shows the change in 2-fox-phenol and propan-1-ol, which had been raised in IBD at baseline, and what happened in IBD after 3 months of treatment. As you can see, a phenol on the left, the raised baseline value in IBD group in green compared with control group in red, fell in follow-up when IBD active and fell lower when IBD was in remission. The differences between baseline and the remission was statistical significance with this p-value corrected with multiple comparison. And similar finding for propan-1-ol shown on the right. Then we compared if there is any difference in fecal focus between disease subtype, Crohn's disease, and ulcerative colitis. Again, the diagram on the left is similar format to the previous one that I show you. This plot shows the ulcerative colitis patient shown in blue triangle and Crohn's disease in red circle. If you look at the loading score on the right, you can see compounds going to the left of the diagram are those associated with Crohn's disease, whereas compounds going to the right of the diagram associated with ulcerative colitis. The statistical model to generate these data has an error rate about 40%, which indicate fair performance. In summary, carboxylic acid, ketones, and aldehyde were lower in abundance in IBD versus control. This may indicate lipid peroxidation as a result of mucosal inflammation. Propan-1-ol and phenol were higher in IBD at baseline and returned to control levels after treatment. Both compounds are end product of amino acid fermentation, so altered level of these FOX may indicate abnormal amino acid metabolism in pre-treatment IBD, and also indicate that FOX may be useful for monitoring response to treatment. Next steps, our findings need to be confirmed and follow-up study. We also evaluating the measurement of FOX in the initial diagnosis of IBD, but this would be by looking at the overall pattern recognition of the FOX profile using the GC sensor, in other words, an electronic nose, rather than the individual FOX measured by GCMS analysis that we show here. I'd like to thank children who participate in this study. We acknowledge our source of funding, including NIHR and Wellcome Trust. I provided some references here that underlie this work, and thank you for listening.
Great, thank you very much, Salma. That was another really fascinating talk. I have a quick question, if that's okay. So, currently, what's the method of diagnosis for these conditions? So if I understand correctly, you know, these volatile compounds offer a sort of a way of diagnosing perhaps sort of more easily or more quickly. What's the current method of diagnosis?
Actually, we collect the stored sample and analyze it by special machine called GCMS. This is machine looked at individual compounds and which is driven actually we have two methods to analyze the data, GCMS and a GC sensor. Actually GCMS which is GC sensor is electronic nose. This machine is very sensitive and is looked at the rapid diagnostic device which built in this kind of observation. Additionally, it is able to diagnose IBD from control with a great accuracy. So the GC sensor has a great ability to differentiate case control, and we will report the data soon. So actually, we're still working on that.
Cool, thank you. So there's a few questions in the chat as well. So the first one says, well done, Salma. Did you also take a history by interview for duration of breastfeeding or use of artificial milks during their earlier childhood?
Yes, so actually we have some demographic data and environmental factors might also affect the FOX primarily through potential affecting gut microbiome. So we collected the data on those such as age, size, sex, pets at household, diet, breastfeeding, and we will look at those factors. But what we are know already, these factors are very similar between cases and control, but we're still working on this.
Okay, thanks. The follow-up question is, thank you, Selma. What are the prospects for turning this into a diagnostic tool? How feasible is it?
Actually, for GCMS data may provide data to develop a biomarker to help differentiate between IBD and other gastrointestinal disease. And also it might be used in clinical laboratory if there are hardware, and these data provide insight pathogenesis, and it is like a border or a novel presentation, and also for therapeutic intervention.
Cool. Thank you very much. That was really interesting. Thanks. And thanks so much for stepping in at such short notice.
Okay, thank you. Bye.
Right, so next we have Huanyuan Luo, who will be talking about risk factors for severe COVID-19. We don't have Huan Yuan, with us today, so we'll be playing a pre-recording, and unless we can, unless they appear before the end of that, then we won't be able to have a Q&A. But do pop your questions in just in case. And in the meantime, here is the talk on COVID-19 risk factors.
Hello everyone, I'm Huan Yuan. My supervisor is Professor Dora Wang from the Department of Clinical Sciences. I'll give a talk on disease progression in patients with COVID-19. The study has been published in a journal as indicated by the link at the bottom. I will give the talk from the following aspects. A previous study in patients with COVID-19 in Wuhan, China, found that the mortality of severe and critically ill patients was 22% and 78% respectively. However, the pattern of disease progression from moderate or less status to severe or critically ill status and the associated factors has not been fully investigated in patients with COVID-19. Assessment of the pattern of disease progression and the identification of associated factors could help physicians to prospectively recognize patients at high risk of progression and help patients to avoid a crisis phase. So the study aimed to describe the occurrence of disease progression in patients with COVID-19 and explore the factors associated with progression. This was a retrospective multi-center cohort study. The study subjects were from 10 January to 29 February 2020, all cases diagnosed with COVID-19 at 24 hospitals in Jiangsu Province, China. The primary outcome was the progression from asymptomatic or mild or moderate status into severe or critically ill status during 14 days follow-up. The study included 625 cases who had complete medical record. No deaths were reported during the study. The median age was 46 years old, and about 50% were male. Table S1 shows the dynamic patterns of disease progression by follow-up day. On admission, that is day 1, about 109 patients, that is about 20% patients, were asymptomatic or had mild status. 488 patients, that is about 80% patients, had moderate status, and 28 patients were severely or critically ill. Overall, changes in disease severity from admission showed an increased proportion of moderate cases deteriorating into severe or critically ill cases, with 1.6% at day 2, progressively increasing up to 5% on 2 at day 7. Table 1 presents disease progression in severity from admission to the worst severity during the 2-week follow-up. Of the 625 patients, 80% had a stable condition or became better during the 14 days hospitalization, while 60% patients progressed by at least 1 degree in disease severity, and 6% patients had progression from day 2 to day 14 after admission, that is, from a symptomatic or mild or moderate status on admission to severe or critically ill status. Table 2 shows the demographic and clinical characteristics of the patients. Compared to patients without progression, the patients with progression were significantly older, more likely to be imported cases who had a contact history with the pandemic center in Wuhan, and more likely to have prior histories of hypertension and diabetes, more likely to have lower mean SpO2, that is, oxygen saturation, and more likely to have a higher median CT quadrant score and higher median proportion of pulmonary opacity volume. And patients with disease progression also had significantly lower median lymphocyte count, median platelet count, and higher median levels of C-reactive protein and median fibrinogen levels. 11 variables were selected for univariate and multivariate analysis. From multivariate analysis, 4 variables measured at admission were identified to be independently related to the occurrence of disease progression, that is, age, pulmonary opacity score, lymphocyte count, and important cases, that is, exposure to the pandemic center in Wuhan. Table S2 shows the oxygen was delivered to patients with the methods shown here. In particular, ventilatory support was used in about 50% of patients with disease progression. In conclusion, Jiangsu province reported low deaths. This was mainly due to the early recognition and early intervention of high-risk and critically ill patients. Among the patients who were asymptomatic or had mild or moderate disease on admission, 6% experienced disease progression to become severe or critically ill. Age, pulmonary opacity score, lymphocyte count on admission, and exposure to the pandemic center in Wuhan were identified as independent risk factors for disease progression. So, the careful attention to those risk factors may help get clinical care. That is my talk. Thank you very much.
Thank you very much. Another really great talk. And we actually have Huan Yuan in the studio with us. If you have any questions for her, please do put them in the Q&A. There's some really intriguing findings there. What do you think is the link with the imported cases or the exposure to the center in Wuhan? Do you think it was a different strain of the virus, or do you think it's to do with how early the symptoms were detected, or what do you think was going on there?
Well, I think for this problem, there may several explanations. As you said, maybe it was at the very start of the pandemic. So those patients were delayed for diagnosis, and so their disease may be more serious and severe. And secondly, so they may have not been be treated with the or be treated by the or be prevented by the some strategies. And also maybe there are different virus variants. Maybe they are different. Viruses were different. But the most important thing, I think, is because it was at the very beginning, so they were not recognized.
Cool, thank you. There is a question from the audience. How easy was it for you to collect the data? Do you think that disease progression has changed now that we have better treatment and more experience with disease progression?
Well, actually, this data it was not collected by me. I was collaborated with a research team in China, and the whole province data were there. So we collected. I did they collected the data while I have done the other things like the study design and data analysis and the manuscript preparation. That's it. Yeah.
Cool, thanks. And do you think that disease progression has changed now that we have better treatment and more experience?
Oh yeah. This could be changed, but I can't say just whether it is changed a lot. We need data to say it. Maybe, yeah, I think so.
Cool. Thank you very much, Huan Yuan. That was really, really cool. Really interesting. Thank you.
Thank you very much.
So yeah, again, another wonderful talk. And our final talk in the session will be by Sepide Saleh, who will be talking about his work on air pollution in rural Malawi.
Hi, so I'm Sepide Saleh. I'm a Wellcome Trust Clinical PhD Fellow, currently working in Malawi, and I'm going to talk about my study Pamodzi, which means together in Chichewa, which is the Malawian language. So just a little bit of background now about air pollution and health. Air pollution is really important in terms of causing morbidity and mortality worldwide. It's the #1 environmental health risk factor worldwide. But the sources of this pollution vary by context. So in Malawi, which is largely rural, cooking with biomass is still a really important cause of exposure, although there are other sources. Also, just to mention the links between air quality and climate. So action on air pollution can have important co-benefits for the environment, vice versa. And also, we know that these airborne pollutants can drive climate change, so it really is a critical health determinant. So I'm going to be talking mainly about the first phase of my study, which aimed through ethnography to gain a comprehensive understanding of air pollution within the social and environmental context. So I used quantitative and qualitative methods, which I'll talk about in a minute. And in addition, I went on to co-develop a whole village clean air intervention with members of the community that I worked in the village. So just talk about a minute about ethnographic work. The mainstay of this was immersive in-person participant observation. So I spent days and evenings, mornings with communities, doing things alongside them and aiming to understand how smoke sits within the context of their day-to-day lives. And this awful picture here just demonstrates me serving the food that I've helped cook with communities. So we did that on a daily basis over an extended period. We also did quantitative air quality monitoring, looking at individual carbon monoxide and fine particulate matter exposures within the village context. And then we did interviews and participatory workshops, which gave us different perspectives on this research question. So this diagram just helps to conceptualize how the different methods sat together. So after initial engagement period, we spent time in each of 6 households doing this in-depth participant observation and concurrent air quality monitoring. At the end of each of these, we had a period of an interview with the household head. And then toward the end, we then did more generic village-based observations and discussions with people. And then we had participatory workshops with village residents toward the end again of this 7-month period. So just a word about the air quality monitoring. This was in 2 sets. So the initial data set involved myself and Henry, my research assistant, doing fine particulate monitoring throughout our ethnographic period. So this white monitor here is the purple air monitor that we used. And we paired this information with detailed field notes. So we were able not only to describe what the source of air pollution were at any given time, but also to provide in-depth data, for example, where it was taking place, what fuel was being used, and what other things were going on. And then after this, we then had an extended data set. So we then added carbon monoxide monitoring. So this is the smaller monitor here. And we did a more standardized set of 24-hour traces on a range of participants. And this picture is Henry, my research assistant, modeling how the monitors were worn. So we're still analyzing the extended data set, but our preliminary results were quite interesting. And I just want to briefly present some of them here. So the main thing is that we had this baseline and peaks pattern. So just to go on here, this is a baseline and peaks pattern. It shows that there were a baseline where there was no identified combustion source going on, and then there were peaks usually related to cooking activity. Interestingly, all across the board, the fine particulate monitor levels were very high. So whereas the WHO recommends a 24-hour average of below 25 micrograms per meter cubed, we were finding that the baseline without any peaks was around 35 micrograms per meter cubed. And then we had peaks going high above that into the thousands. So when we look again at the baseline and peaks pattern, this shows a paired traces between, on top, the female in the household who was doing the cooking, and the bottom one is the male in the same household who tends to sit a little bit further away and do other things. And we see that whereas the baselines are similar, the peaks in the female trace go up in the thousands up to 4,000 or 5,000, and in the male trace only went up into the hundreds. So on this preliminary data set, we also did a bit of regression to look at things like location, which determines ventilation level, cooking device and fuel, and how these impacted the overall exposures. And the regression essentially showed that the combination of things associated with the lowest cooking related PM2.5 levels were cook stoves rather than three-stone fires, and these used in well-ventilated spaces, so for example outside or on a veranda. Just going on to the ethnographic themes now, the qualitative themes coming out from the participant observations and other things. So we found that cooking as an exposure is gendered, almost always women doing the cooking, and it's culturally shaped. But in Malawi, we found that economic scarcity throughout this setting was a major constraint to clean air. And this worked through 3 pathways. So firstly, limitations on choice. And you see this quote in red here, just shows that women were using, for example, reed mats as they didn't have any other fuel to use. And it was very smoky, but there was no option, there was no other thing, and people had to feed their families and eat. Secondly, there was a sense of this day-to-day hardship throughout the setting. So the quote in green shows that people were aware, yes, it did hurt their eyes, but this wasn't important in terms of having to get cooking done. And in terms of the wider difficulties, so that includes cooking sema, which is a really quite hard physically to do in terms of getting water in the boiling heat and taking maize to the maize mill. So really this small discomfort wasn't significant. And the third thing that was really important was the sense of precarity. So life is precarious. And this picture here shows it. This is a household that we spent a lot of time with just before Christmas. And they'd spent a lot of time and effort saving up and building this standalone kitchen building. And it was all nice and new just before we left before Christmas. And then we came back in January and it had been washed away, as you can see, by the floods. And there is a sense generally throughout that nothing is permanent, nothing is constant. So you could spend a lot of time building up to something or a lot of energy on something, and it could be taken away the next minute, and that goes for health as well. Just spending a minute on the participatory workshop findings. A lot of the themes from the participant observation came up here as well. And when we talked about potential interventions for clean air, we found that scarcity severely restricted any possibilities, any interventions that we could think of. Interestingly, something that came up a lot was what participants said, business. And that essentially meant to them just getting an ad hoc work, any sort of job to allow them to have more of an income, and that would help them to buy stoves or charcoal. So in terms of all of this, our intervention plan was modest, it was village-wide and something with a high possibility of sustainability. So we went for a locally made firewood cook stove, and this is it. It's a ceramic cook stove called Chitete Zombala, which means protecting cook stove, and it's very cheap. Quite modest, but it's something that people could buy themselves subsequently. And then we coupled this with cooking outdoors or on a condi where possible to try and bring exposures down even more. And this is something that we're now trialing throughout the village, and we're looking at how it's going. Currently, it's been taken up really, really well in the village. So I've finished a little early. This is a picture of my cat, Mackie, who came back from the village with me one day and has stayed. And he's on the monitor bags, which he particularly likes because they smell very smoky. I'm happy to answer any questions that anyone might have or talk about any issues. Thank you.
Brilliant, thank you very much. Really interesting, so we're getting some feedback. I'm not sure why. So I don't think there are any questions in the Q&A yet. I just wanted to ask, is there sort of an appreciation of the sort of long-term negative effects of this smoke? Is it sort of perceived as an inconvenience, like right now this is really unpleasant, or is there an understanding that, you know, long-term there are going to be problems as well?
Yeah, so when we came, there weren't people didn't really know about the long-term effects of smoke. So that was something that we brought, and we really wanted to bring people's perceptions together. So we learned from people and they learned from us. But as I say, in terms of the qualitative themes, there was a sense that even when people knew that, there was a lot that they were contending with that meant it was very difficult for them to do anything long-term, really. So yeah, they did learn and they tried, but it's quite difficult.
Yeah, because I guess the stove, you know, you still need the fuel for it, and if you're burning things like reed mat, then I don't know, I don't know to what extent that the stove can accommodate, you know, just sort of haphazard fuel that you find that it's the only thing you can find, and if it can accommodate it, you know, does it then still reduce the emissions when you burn that kind of fuel inside it?
Yeah, that's really interesting. Yeah, so actually when people get to the point where they're burning things like clothes and reed mats, then yep, stove's not so great. But it works really well for fuels just picked up off the ground, and even for things like maize cobs and maize stalks that are used a lot. I don't think it will reduce exposures a lot, but it's a lot more efficient in its burning. So people are really liking using it and using it across the village.
Cool, thanks very much. There's a question from the audience as well. Did you include children in the exposure monitoring?
Yeah, thanks for that. It's a good question. So initially we used myself and Henry as proxies for the people in the household because we were doing the same things as people in the household. And then in the second stage, we just used a set of adults. So we didn't, partly because the monitors, even though they're relatively small nowadays, they're quite chunky to be carried around the waist, and also children do a lot of stuff which could potentially damage the monitors, and we didn't want to put that sort of thing on the children. But a lot of the ambient monitoring that we're doing would reflect children's exposures.
Cool, thank you very much. So yeah, no, that's another really interesting talk to finish what has been, I think, a really, really good session with some really nice diversity of topics and really good speakers all around. So thank you, Sapida, and thank you to all of our speakers for that session. So that takes us to the end of the first session, which I think has made a really great start to the conference. Please do visit the posters and the breakout rooms when you get the opportunity. We're now just taking a quick 10-minute comfort break before the next session. But from me, that's it. And I'd just like to say, I hope you enjoyed that first session, and please enjoy the rest of the conference.
Good morning. Can everyone hear me okay? Welcome to session 2. We've got 5 talks lined up that should be quite interesting. The first one we're going to start with is Charlotte Hemingway, looking at gaming in HIV.
Hello everyone, and so today I will be presenting findings from a qualitative study which formed part of my PhD. And the overarching aim of my project was to better understand the potential of digital games within the context of HIV prevention in the Philippines. And the game, titled Battle in the Blood, which you can see in the video, was launched on the app stores in December 2017. But first, I want to take you back to 2016 and the public health challenge that inspired this work. So the Philippines has the fastest growing HIV epidemics in the region, with cases forecast to triple in the next 10 years, and with a majority of new infections among 15 to 24-year-olds. There was this clear and urgent need to increase demand and access to HIV services within this age group, in order to slow the rate of transmission and prevent that forecast from becoming a reality. So in the Philippines, increased online connectivity, a growing economy, and prevalent mobile device use has changed the way people spend their time and socialize, placing mobile games as a promising tool to reach young key populations. And the idea to use digital games to promote HIV services was first conceived by Dr. Emmanuel Baja back in 2015. Dr. Baja became aware of a series of HIV cases among young men who were selling themselves to fund their online gaming habit. And this was later included as one of the short stories in the game. So from this observation and knowledge of the HIV epidemic, it was theorized that a mobile game could provide an effective means to advocate HIV services and reduce behavior. So the game design process was divided into 4 steps and informed by relevant theory. Design workshops were conducted with target users. Formative research on enablers and barriers to HIV services were translated into a set of learning objectives, and then they were mapped to mechanics and features in the game. And lastly, I developed a conceptual framework to express how the game might achieve its target outcome. So the target behavioral outcome was defined as undergoing HIV testing and counselling from a legitimate provider when the individual perceives they are at risk, with timely linkage to care if reactive. And the framework is based on Fishbein's integrated model of behaviour prediction. It makes the assumption that the game's content will directly affect salient beliefs through the learning objectives, which in turn influences key cognitive mediators of behaviour. However, the ability for the user to act on new intentions would still remain dependent on external conditions which the game may have no influence. The project consisted of 3 interlinked studies. Outcomes from the first study were used to inform the game design. Outcomes from the second study, referred to as the game development phase, were used to determine the game's acceptability. And this was done using a combination of prototype testing sessions, which you can see pictured, and game analytics. Information was also used to make improvements to the game. In summary, the game was found to be highly acceptable, and when played in a facilitated environment, had a positive effect on the reported knowledge and attitudes. The game analytics demonstrated high uptake of the game among target users, but also reported that only a small proportion had completed the game, suggesting that the gameplay had limited appeal. To make best use of the limited time, I will focus my presentation on the third study, where I explored the impact of the game from the perspective of the end user. And this was conducted to better understand the mechanisms in which the game influenced HIV-related behaviour, if it did so at all. I also included a 5th objective on the user's understanding of data collected through the game, as previous studies have found that terms of service policy are widely ignored by the public, and it's likely the same would be true for Battle in the Blood. So data for the study was collected using semi-structured interviews with end users. I used a combination of inductive and deductive approaches to the analysis. So while the exploration was guided by a predetermined conceptual framework, a solely deductive approach could preclude the development of new theory to better explain how the game affects behavior. Interviews were conducted face-to-face in public cafes in secluded areas, and this was done in the project regions, which you can see pictured. 25 game users were recruited. 5 participants from Davao disclosed their HIV positive status at their own discretion, and 1 was diagnosed after discovering and playing the game. The studies found that uptake of the game was primarily driven through word of mouth, and the ability to play the game anytime for free without the need of an internet connection was a dominant factor in continued game use. Participants in the National Capital Region, where the traffic is notorious, often played the game while commuting. The game appeared successful in increasing awareness among users of the availability and the benefits of HIV treatment, and also instilled a sense of hope that HIV is not a death sentence and life can go on. Participants also demonstrated increased awareness of the benefits of early access to HIV treatment. Learning was predominantly achieved through interpretation of the game's narrative, and practical knowledge on HIV services, such as the location of clinics, was almost never obtained through interaction with the game, as access to this information required an internet connection, which was not compatible with the situations users typically played the game. So of the 7 participants who had never been tested prior to playing the game, 3, all heterosexual, did not perceive themselves to be at risk and had no intention to engage in testing services. 4 reported that the game had encouraged them to seek out testing services, but only 1 had acted on his intentions at the time of the interview, and this is who is quoted on the slide. In this case, the game provided the tipping point in his decision to access HIV testing and treatment services. In the one instance where the game triggered testing behavior, the meaning the participant derived from the game's narrative and the impact on his attitude towards testing was shaped by the recent loss of his partner. And the interviews also provided evidence of the game being used as a jovial and reputable mechanism to encourage others to test and introduce the topic of HIV to friends and family. So overall, the game appeared to have a limited effect on the socially ingrained habits, dispositions, and beliefs of the participants. While the game was successful in influencing cognitive factors of HIV service uptake, the ability for the users to act on these thought processes remained dependent on external social conditions. The interviews also demonstrated that positive action was taken if the user encountered a character or a storyline to which they could directly relate to, or recognize key commonalities with their current situation or social group. However, the participants also believed that the game lacked something in its design to make it go viral, increasing its impact on the wider community by delivering something that would really get people talking. So from this understanding, I revised the conceptual framework. In the revised framework, potential game influence is encompassed by the level of enjoyment and narrative relatability experienced by the player. The interplay between the impact of the game and the participant's social reality is reflected through the inclusion of social field habitus and doxa under modifying factors, and this encompasses both the individual perceptions and behavior. So where the behavior change game operates at the individual level, as is the apparent case for Battle in the Bud, its potential effect is limited without the necessary conditions for behavior change at the spatial level or social field. And then finally, for objective 5, it was evident from the interviews that none of the participants had accessed the game's privacy policy or App Store description, which provides details on how data collected by the game is managed. But despite this, most participants demonstrated an accurate understanding of the purpose of the game analytics. However, there were some misunderstandings, with one participant suggesting that it was used to spy on players and monitor their sexual behavior. So from this project, I formed a set of recommendations for the design and development of behavior change games, which take into account the importance of the bounded context around the game. from the composition and dynamics of the game development team to the interplay between the user's social reality and the impact of the game on their behavior. Now, digital gameplay as an activity does not occur in a vacuum. Users talk about games, share strategies, and participate in a gaming community, which can span multiple media platforms. And social interactions triggered through the gameplay and the user-centered design process may provide opportunities beyond the game's content to effectively influence and change behavior. And this should be considered in those early design stages for the game. And when the target behavioral goal for the game is dependent on conditions in the health system, it's important to think of the game as a tool within the wider health system strengthening intervention in order to maximize its potential. And because of this, it may also be beneficial to utilize case study methodology to evaluate the game. And lastly, I also discussed the use of game analytics in games for health research and how ethical concerns identified during the study present an opportunity to innovate in how users are informed and give consent. So I'd just like to finish by thanking my colleagues at the University of the Philippines, my supervisors and mentors, and to all the people at the Community Health Systems Group and EM Studios. And lastly, but not least, to all of the participants who braved the infamous Manila traffic to meet with me and share their stories. Thank you for listening.
Thank you very much. That was really interesting. This isn't my area, but are there any other behavioural change games that have had success in different fields?
It's a mixed bag, and I think when you look at the meta-analysis of game effectiveness, we're seeing quite consistently small effect sizes or no effect at all. So I think that is potentially down to two possible reasons. One is that games are not that effective at influencing behavior, but also it may be that randomized control trials are not sensitive to game effect. So we might also want to look at exploring alternative ways to evaluate these tools. And I think an important part of that is the fact that there is potential to influence and change behavior outside of the content of the game, which is why a randomized control trial might not be appropriate.
How would you measure that then?
So this is where I was thinking around case study methodologies. So potentially looking at using data from multiple sources to explore and evaluate the case under question, which would be the game. So looking at the game within the context of its use.
Brilliant. Thank you very much. That was very interesting. Right, so our second talk is by Katerina, and it's developing a novel multiplex assay for pneumococcal disease.
Hi everyone. Today I'll be talking to you about how I've developed a novel multiplex assay using Luminex technology to screen for pneumococcal vaccine candidates. So pneumonia is the leading infectious cause of death in children under 5 worldwide. And in fact, 1 child dies every 2 minutes from pneumonia. Over 90% of deaths occur in sub-Saharan Africa and Asia, but it's a major cause of morbidity and mortality worldwide in both high and low income countries. So the majority of cases are caused by the bacteria Streptococcus pneumoniae. Over 2/3 of pneumonia cases are caused by the bacteria. In 2016 alone, pneumococcal pneumonia resulted in a total of around 1.2 million deaths across all ages, and the disease accounts for approximately 200 million cases per year. So risk factors for the disease are age, so predominantly it affects children under 5 years of age and adults over 65 years of age, concomitant viral infection, heavy drinking and smoking, exposure to air pollution, and chronic disease such as chronic lung disease, diabetes, and renal disease. So there are over 94 different capsular serotypes of the bacteria. The current vaccines we have do not protect against all of these serotypes. So we have two vaccines that are used. We have Prevnar 13 and Pneumovax 23, and they protect against 13 and 23 serotypes respectively. With the Pneumovax vaccine, this is routinely given to adults over 65 years of age, but it's not immunogenic in children under 2, because until they are over 2 years age, they don't mount serotype-specific antibody responses. And while this vaccine is good at protecting against invasive pneumococcal disease, we believe that it is not very good at protecting against non-bacteremic pneumonia. And we see quite a similar sort of issue with Prevnar 13. So while the efficacy against invasive pneumococcal disease is around 70 to 97% for non-bacteremic pneumonia, it's a lot less, around 17 to 37%. There are also other drawbacks with these vaccines, the main one being serotype replacement. So if you look at this graph on the right, you see that with the introduction of vaccines, the prevalence of vaccine type serotypes decreases, but we then get an increase in non-vaccine serotype prevalence. In addition, the vaccine formulations are based on serotypes most prevalent in high-income countries as opposed to low-income countries where the burden of disease is highest. And so there is definitely a need for improved vaccines which have universal coverage against all pneumococcal serotypes. And one way we may be able to achieve this is through protein vaccines which have a different mechanism of action. However, these protein vaccines do not yet exist against pneumococcal pneumonia, which is where my project comes in. So just to give you a bit of background, in our group we have the experimental human pneumococcal carriage model. And pneumococcus is part of the natural flora. So at any one time, around 10% of adults and around 40 to 60% of children will be colonized with pneumococcus. And what we do is we recruit volunteers and we intranasally inoculate them with pneumococcus. In general, around 50% of our participants will develop carriage and 50% will be naturally protected against carriage. And we take samples at baseline and then post-challenge, generally around 14 to 29 days post-challenge. So my research questions were which pneumococcal protein antigens are protective against pneumococcal colonization and which pneumococcal protein antigens are immunogenic. So what we might expect is that in the volunteers who are naturally protected against colonization, they have higher antibody levels at baseline to certain pneumococcal proteins. We also know that colonization with pneumococcus mounts an antibody response and an immune response that may protect against recolonization in the future. So what I did, I had a library of 74 purified pneumococcal proteins. This is the largest protein library of its kind in the world. So all of these proteins are in their natural conformation. They're all surface exposed and they are conserved across pneumococcal serotypes. And this library was developed by collaborators in Harvard University. And I have microspheres and I conjugate these antigens to those microspheres. So each of these different microspheres is impregnated with a different fluorophore that fluoresces at a different region of the visible spectrum when exposed to lasers. I have 22 different regions, which means that I can develop an assay where I can measure antibody response for up to 22 different antigens at a time. And what I do is I incubate these microspheres conjugated to the antigens with serum samples taken from our EHPC volunteers. Antibodies in that serum will then bind to those antigens. And then, much like an ELISA, I then add detection antibodies, and level of fluorescence indicates level of antibody binding. So in this way, I can quantify levels of antigen-specific binding. So just to give you an overview, the assay was optimized in a number of different ways. So firstly, it was important for me to optimize the assay for a known immunogenic pneumococcal protein. So in this way, if I wasn't seeing any response, it's probably because the assay isn't working rather than because it isn't there, because we know that these proteins are immunogenic. So I looked at optimal concentration of secondary antibody, serum concentration, and incubation period. I then had to verify that the microspheres of different regions give comparable MFI readings for the same protein. And as I say, I have 22 different regions. So of the 74 proteins, they were all sort of allocated to across 4 different panels randomly. I then repeated the optimization with 4 further pneumococcal proteins, some highly immunogenic, some less immunogenic, to ensure that the assay optimized conditions were the same for the different proteins, and then ensure good correlation between results when running singleplex and multiplex, and then evaluate the median intra and inter-assay variation to make sure that it's comparable against assays, like similar assays that have been developed. So I have 2 different cohorts of volunteers. I have 24 volunteers that were inoculated with serotype 15B pneumococcus and 39 volunteers inoculated with serotype 6B pneumococcus. Of those inoculated with 15B, 15 were naturally protected against colonization and 9 developed carriage. In the 6B cohort, 18 were naturally protected and 21 developed carriage. Importantly, in the 15B cohort, serum samples were taken prior to inoculation and then 14 days post-inoculation, whereas in the 6B cohort, samples were taken 4 days prior to inoculation and then 29 days post-inoculation. So as I say, I then ran these serum samples in my newly developed assay. And what I saw, so this is just, these graphs are for each panel of proteins that I had. There was no significant difference in humoral response between protected and susceptible cohorts in either the 15B or the 6B cohort. However, colonization did induce protein-specific humoral response. So we saw that antibody levels were increased to the majority of proteins post-colonization with pneumococcus. But this was only significant in the 6B cohort. So the serotype 6B pneumococcus elicits a much greater immunogenicity than 15B. Overall, I identified 5 novel protein vaccine candidates. So this was following Benjamini-Hochberg correction. And as I say, there were no proteins that elicited significantly higher antibody levels post-colonization with 15B as compared to before challenge, but 5 proteins elicited significantly higher antibody levels post-colonization with 6B as compared to before challenge. And at least 4 of these proteins show good levels of immunogenicity. However, there was no correlation with colonization density. So the next steps, I'm going to take these candidates forward to run B-cell ELISpots with proteins that show increased antibody binding post-colonization. And yes, I would just like to thank all of my supervisors and Eliani and Richard Malley, who are our collaborators who provided the proteins for this, and all of the EHPC volunteers without whom none of this work would be possible. Thank you very much.
Thank you. That was really interesting. A quick question from me, and I might have missed this. What was the rationale for using the 2 different strains of pneumococcus, the 15B and the 6B?
It was basically to see if one strain elicited more protection than the other. Sorry, my screen stopped sharing. And also to see if one strain was more immunogenic than the other, but basically I had access to these 2 different cohorts of people. I could have just done it with one, but I thought it would be interesting to do it with both to see if there were certain proteins that were coming up when people were inoculated with different strains.
Is there anything special about those 2 strains, or are they just happen to be ones you've got, or are they particularly virulent? Or
so the 6B strain is the one that we use predominantly in the EHPC model, and it's a strain that we know causes a lot of disease worldwide. It's also a really good colonizer of the nasopharynx, which is what we want in the model. 15B likewise, but we found that it is less effective at colonizing.
Brilliant. Thank you very much.
Thank you.
And I'll just hold and see if there's any questions from the audience. Don't think so, but thank you. That was really interesting. Right. The next up is Wissam, who's talking about I can't say it, Plasmodium falciparum gametocytes. Sorry about that.
Hi. Okay. So the sound is okay? Hi, my name is Wissam Bakhsh. I'm going to talk about pharmacodynamics of Plasmodium falciparum gametocytes. And as a background, Plasmodium falciparum has a complex life cycle, consists of asexual stages are responsible for clinical manifestation, and sexual stages are responsible for transmission of the parasite from human to the mosquito through the blood meal and transmission to a new host. 6 ratio and gametocyte density could affect transmission to the vector. Also, drugs have been shown to differentially affect male and female gametocyte. As a primary aim of this project is to gain a deeper understanding of the pharmacodynamics of late-stage gametocytes. As a method, NF54 strain of Plasmodium falciparum was induced to produce gametocytes. Then, following magnetic purification and cell suspension, was stained with cypergreen and mitotricker debrid to identify DNA and membrane potential, respectively. Then used fluorescent activated cell sorting to sort uninfected red blood cells, asexual stages, and sexual stages. Finally, sorted cell populations were assessed by dim system microscopic examination and confocal microscopy. The first objective is to develop a robust and reproducible assay that to determination of gametocyte viability. We have developed a robust flow cytometry assay that uses cybergreen to indiscriminately stain DNA in viable and dead parasite, and mitotricker debrid to selectively stain mitochondria in viable parasite. For doing that, mixed culture of Plasmodium falciparum was stained with cybergreen and mitotricker and showed one peak on the negative and 2 peaks on the positive side of cybergreen and mitotricker histograms. And by sorting these population, uninfected red blood cells were found in B1, asexual stages were found in B2, whereas B3 was represent gametocyte population. Then flow cytometry analysis of gametocyte culture showed 2 positive peaks on cybergreen and mitotricker histogram. B1 was corresponding to low fluorescent intensity signal, includes asexual stages and dying gametocytes, whereas P2 were corresponding to late stage gametocyte. Then, to discriminate late stage mature male and female gametocytes, I have targeted alpha tubulin protein expressed in mature male gametocytes by using primary antibody and conjugated with secondary antibody labeled with Alexaflor 405. Late stage gametocyte culture was gated based on forward scattered area and side scattered area. Then uninfected red blood cells were gated out by selecting positive population on cybergreen histogram. Dying and dead parasite were gated out by selecting positive population on mitotricker histogram. Then finally, the remaining gametocyte population was gated based on a combination of mitotricker debrid and Alexaflor 405 in order to discriminate between mature male and female gametocytes. Late stage gametocyte viability was assessed by flow cytometry using mitochondrial dye and gametocyte culture treated with FCCP FCCP methylene blue and compared to untreated sample. So the sample treated with FCCP showed a reduction in fluorescent intensity signal, but those untreated and the sample treated with methylene blue didn't show a comparable effect. So we did observe that this result was consistent with confocal microscopy, as we can see that the sample treated with methylene blue showed bright red fluorescent signal indicate to unharmed mitochondria as well as an energized parasite plasma membrane and consequently a viable parasite, whereas the sample treated with FCCP didn't show, the green fluorescent signal and without very bright red ones indicating to dead or dying gametocyte as we can see in the sample treated with FCCP. So the next objective is to further developed the assay in order to distinguish the viability of mature male and female gametocyte when treated with antimalarial agent. For that, gametocyte culture treated singly or in combination with a defined group of antimalarial agents such as atovaquone, concannamycin, methylene blue, and FCCB. The untreated sample was considered as a maximum fluorescent intensity signal at 100%, and the sample treated with FCCB was considered as a minimum fluorescent intensity signal at 0%, and that was considered as a dynamic range. The results showed that showed an effect on gametocyte membrane potential by atovaquone alone when combined with concannamycin, and that was collapsed the mitochondrial membrane potential of male and female gametocytes compared to untreated sample. Then the next objective is to gain a deeper understanding of mechanism and processes underpinning the differential response of male and female gametocyte to drugs. I have evaluated the difference between male and female gametocyte based on some physiological differences such as mitochondrial membrane potential, thiol level, and reactive oxygen species in the form of mitochondrial superoxide indicated by enhanced mitotrichard bread, monochlorobiomin, and mitochondrial superoxide dyes respectively, and the results showed that male gametocyte population demonstrated a higher mitochondrial membrane potential as well as thiol level compared to female gametocyte population, whereas in mitochondrial superoxide there was no difference in both population. In order to distinguish, to investigate the different response of male and female gametocytes to drug based on the thiol level, the fluorescent signal was expressed by thiol and monochlorobiomin adduct assist and was higher among male gametocyte population compared to the female ones in untreated sample. Then, in order to assess the effect of drug in thiol level, the culture was treated with methylene blue and atovaquone, and the sample treated with methylene blue showed an increase in the fluorescent intensity signal in male gametocyte population compared to untreated sample. To investigate the different response of male and female gametocyte to drug based on reactive oxygen species in the form of mitochondrial superoxide generated as a byproduct of oxidation process. The fluorescent signal exhibited when mitosox reagent is oxidized by superoxide in mitochondria, and the result showed that there was no difference between male and female gametocytes in mitochondrial superoxide in untreated sample as well as in the sample treated with in untreated sample and as well as when treated with antimalarial agent. As a conclusion, flow cytometry using cybergreen and mitotricker is a sensitive and highly reproducible method that is able to identify and isolate mature gametocyte, also to check the viability of gametocytes. Currently, we are trying to complete the physiological differences between male and female gametocyte based on pH level in both gender. We will test compounds that are specific to male and female gametocyte and characterize the specific target of this compound. Thank you.
Thank you for a really interesting talk. Thank you very much. You briefly mentioned at the end, but what's the next step for this work?
As I mentioned in the conclusion, I will test compounds that are differentially specific to male and female gametocytes, as the male has a wide range of antimalarial drug target as like a vulnerable target for antimalarial drugs and action, and we will differentiate and we will see the characterize of this compound, target of this compound.
Thank you. Right. Next up, we have Frank McCann talking about the durability of insecticide-treated nets.
Hello everyone, good morning. I'm Frank. I'm a PhD student in the Vector Department, supervised by Lisa Reimer, Martin Donnelly and Philip McCall, and I'm going to be talking to you today about my project, which is the durability of pyrethroid long-lasting insecticidal nets with and without piperona butoxide. So first, a bit of background. So long-lasting insecticidal nets, LLINs, are the cornerstone of malaria control strategy, forming a physical and chemical barrier against the bites of malaria-transmitting mosquitoes. However, their effectiveness is threatened by the widespread rise of pyrethroid resistance, which are the insecticides used in these nets. So in response, LINs have been developed that contain the Syngis piperona butoxide, and this combination of the pyrethroid and PBO restores effectiveness against these resistant mosquitoes, and this new class of net is referred to as PBO LLINs. So the WHO has given an interim endorsement to these PBO LINs to be used in areas of moderate pyrethroid resistance, and have since been demonstrated to be about 30% more effective than standard nets. However, data on their operational lifespan is lacking, meaning that we don't know how long these nets last in the field before they have to be replaced. So the aim of this study is to quantify the physical and chemical integrity of two PBO LLINs across 2 years of operational use, with comparison to their conventional equivalents. There are 3 objectives. First, we have physical integrity, the extent of damage. We have the chemical integrity, the total amount of the chemistry that's remaining, and bioefficacy, so the insecticidal effect on pyrethroid susceptible and pyrethroid resistant mosquitoes. So for the study design, we have 4 different nets that we're assessing. We have Olicet, that contains only the pyrethroid permethrin, Olicet Plus, that contains both permethrin and PBO, Permanent 2, that contains only the pyrethroid deltamethrin, and Permanent 3, that contains both deltamethrin and PBO. These nets were provided from a randomized control trial embedded into Uganda's national LIN distribution. It took place from 2017 to 2018, and nets were sampled at households from 12 and 25 months post-distribution. So objective 1, physical integrity. The methods here are quite straightforward. So the length and width of each hole was measured using a ruler, and then the area of that hole calculated on the assumption it wasn't an ellipse. There are 2 endpoints here. First, we have the proportion of nets in each proportionate hole index category, which is simply we sum up the total of damage on that net and then categorize it as either good, acceptable, or needs replaced based on the bands below. The second objective is to quantify the total area of damage on each sector of the net. So as you can see in the graphic there, we have the top, the upper third, and the lower 2/3. And this is based on previous behavioral work that show that mosquito activity is largely on the top, a small amount of mosquito activity on the upper third, and very little activity on the lower thirds. And then in addition to comparing different types of net, we'll also be comparing these outcomes for traditional versus improved housing. And to clarify what I mean by that, we can see traditional housing on the left, which is these mud-walled straw-roofed houses, and improved housing on the right, which are these brick-walled iron-roofed houses. So onto the results. So for the proportionate hole index, we can see that about after 12 months, 6.6% of nets needed to be replaced, and it's approximately doubled after 2 years. Importantly, there was no difference in the probability of needing replaced between LIN products, meaning that both PBO LINs were just as durable as their standard equivalents by this metric. However, when we look a little closer, we can see some trends appear. So the damage was heavily concentrated on the lower part of the net, closest to the ground, with very little damage on the top of the net. And if we look at a ratio here from the lower up to top, it goes about 10 to 3.7 to 1. However, if you compare traditional and improved housing, we can see there's far greater damage in traditional housing. And in particular, the nets in traditional housing had a lot of damage on that lower part of the net. However, nonetheless, there was no difference in this damage ratio between different LIN products. So given that there was so much more damage to nets in traditional housing, I decided to revisit the categories shown before. And here we can see here that nets from traditional housing were far more likely to need replaced than those from improved housing. And in fact, after 2 years, while only 1 in 10 nets from improved housing needed to be replaced, almost 1 in 3 nets from traditional housing had to be replaced after 2 years. So moving on now to chemical integrity. So the measurement here is the total quantity of the active ingredients remaining at each time point, and this was done with 30 nets of each LIN type at each time point, and was quantified using high-performance liquid chromatography. So the results here. So we can see that pyrethroid content was relatively stable over time, and in fact, after 2 years, Permanent 2, Olicet, and Olicet Plus were not statistically distinguishable from baseline. The only net that did statistically decline was Permanet 3, which declined by about 30% over the 2 years. However, even then, all of these nets were still within the acceptable range of the target dose set by the manufacturers. However, if we look at the chemical content for PBO, it's a very different story. So PBO content declined rapidly for both PBO LANs over that same time, with after 2 years, Olicet Plus had declined by 55% and Permanet 3 declined by 58%. So we've seen these large declines in PBO, but what do these actually mean in practice? As we've only measured the internal chemistry, what we want to know is how much of that insecticide is actually at the surface affecting the mosquito. And that's why we do these bioefficacy assays, where we look at the effect on pyrethroid susceptible and pyrethroid resistant mosquitoes. So the primary method here of assessing bioefficacy is a WHO cone bioassay, which you can see on the picture on the left, where we hold the mosquitoes in a cone against the net. And the nets that perform poorly in this assay were then assessed with a follow-up assay, which is WHO wire ball assay. You can see the picture on the right, where the mosquitoes are held in a cube with a net on all sides. Otherwise, the methods for these two are exactly the same. So Anopheles gambiae SS mosquitoes were put inside for 3 minutes. And then after this was complete, we count the number of mosquitoes knocked down after one hour, and then the number of mosquitoes dead after 24 hours. This is done with 3 to 5 day-old unfed females as per WHO guidelines. And there are 2 different components to this. So we have first, we do this on pyrethroid susceptible mosquitoes, which is Kusumu, and then for the pyrethroid resistant strain, which we call Busia, which was established at LSTM from collections Busia, Uganda, one of the same study sites. And we've since characterized it to have both target size insensitivity and moderate metabolic detoxification. So it's a moderately pyrethroid resistant strain. So on to the results. So first, quickly, for the pyrethroid susceptible strain, as expected, both PBO LANs were fully effective against pyrethroid susceptible mosquitoes at baseline, and there was no difference in knockdown or mortality for any product. So all of these nets both our PBO LANs were fully effective at baseline and fully effective again at 12 months later. However, when we look at for pyrethroid resistant mosquitoes in the cone test, it's quite a different result. So we can see that Permanet 3 was the only net that was fully lethal when it was new, and mortality declined by about just over 25% each year, meaning that after 2 years, less than half mosquitoes were dying. Unexpectedly, mortality with Olicet Plus was very low throughout all the sample time points, which is surprising as it contains both permethrin and PBO, and in fact was no different from the pyrethroid only nets Olicet and Permanet 2, which achieved very low mortality. So given this unexpected result with Olicet Plus, that's why we did these follow-up assays, and here we can see we get a very different result. So this is with the exact same nets in the same strain of mosquitoes, but we see in the wireable assay we have a profoundly different outcome from the cone bioassay. So these assays are telling us that this net is now highly effective against these mosquitoes, achieving 92% knockdown and 86% mortality. So finally, before I wrap up, I want to show what's possible. When you have the chemical content of every net and also the bioefficacy of every net, you can look at the relationship between those 2 things and start making predictions using statistical models of how effective a net would be for a given chemical content. As an example here with Permanet 3, we can see that delta methrin content actually has little impact on mortality against pyrethroid resistant colony, at least within the range of values that we actually see in real life in a study. Instead, bioefficacy was closely linked to PBO content, and in fact we can put a threshold on this and say that you can maintain above 80% mortality as long as you keep at least 15 grams per kilogram of PBO, but if it goes below that point, then bioefficacy would be expected to decline rapidly. So to wrap up with key points and implications, for physical integrity, we can see that there's no difference in physical outcomes between PBO LANs and their pyrethroid-only equivalents. There was very little damage on the top of the net, where most mosquito activity actually occurs, with the vast majority of activity happening close to the ground where we'd expect very little mosquito activity, and this perhaps challenges the assumption of the current guidance that all holes are counted equally no matter where they are. We saw that there was much greater damage to nets in traditional housing, with in fact close to a third of nets needing replaced after 2 years. And this suggests a more targeted approach to net distributions may be prudent, with regions where traditional housing is more commonplace perhaps should be getting nets more frequently. For chemical integrity, we saw that pyrethroid content was fairly stable over time, but PBO declined sharply, and this PBO decline was linked with a drop in bioefficacy. And for the bioefficacy, we can see that both PBO LANs were highly effective against pyrethroid-resistant mosquitoes at baseline. However, the outcome varied depending on the assay, with Olicet Plus performing poorly in the cone, but then was fully effective in the wire ball despite being the exact same nets with the exact same mosquitoes. This suggests that there's something here we don't quite understand, and this should be the subject of further work to work out why these 2 different assays provide such a different result. And then finally, to wrap up, Longitudinally, permanence-free retained high knockdown throughout the 2 years of the trial, but then but over the same time, mortality declined relatively quickly. So that's me done for the day. I'd like to thank very much to my supervisors, Lisa, Martin, and Philip, and also to Amy and Jonathan for all their help with rearing mosquitoes and working and experiments with Exquil, and also to everyone else involved. Thank you very much for listening.
Thank you, Frank. That was really interesting. I have a couple of questions here. What do you think is the reason for the association with housing type? Do you think it's causative or correlative?
I can't hear anything.
Sorry. Can you hear me, Frank? Frank?
Sorry, I can't hear you at all, Amy.
Right, I don't know whether anyone... Can you see the comments? There are some questions, if you can see those. I was asking, what do you think the reason for the association with housing type is? Right, I think we better move on. I think Frank's having some technical difficulties. Sorry about that, everyone, and sorry, Frank. Okay, so we now have Jacqueline talking about point-of-care ultrasound in acutely breathless patients. Thanks, Jacqueline.
Good afternoon, everybody from Nairobi. This is a study which is part of my PhD work, and the topic is on point-of-care ultrasound in acutely breathless patients, this is a qualitative study of the enablers and challenges in a teaching hospital in Kenya. My primary supervisor is Jamie Rylands. Acute breathlessness is a common distressing symptom which is experienced by lots of patients in the emergency department. Point-of-care ultrasound, as I have a picture of it here, is a rapid and focused imaging test which is usually performed at the bedside and not in a radiology department per se. It's non-invasive and has no risk of ionizing radiation. Adoption of this point-of-care ultrasound could promote accurate and early diagnosis and treatment in patients who are acutely breathless, and this may be particularly useful in low-resource settings where we don't have access to gold standard diagnostic testings. The aim of the study was to explore the experience of point-of-care ultrasound used in the emergency department, and to understand the facilitators and constraints of incorporation into the patient investigation pathways. We did data collection involved key informant interviews using semi-structured interview guides, which ran from September 2019 to February 2020. And the participants were purposively sampled and included clinicians, frontline health practitioners, and also mid-level hospital managers who are clinicians. We used the analytical framework approach with concurrent thematic analysis. The framework approach we used was the organizational capacity, which is adapted from Aragon and Giles. And this looks at three aspects. We have the tangible software, the intangible softwares, and the hardwares. And we looked at this from the individual level, hospital level, and also system level. To understand the behavior of the clinicians, we use the capability, opportunity, and motivation behavioral model. Our results showed that the clinicians thought use of the clinical POCA's integrated approach was beneficial, but at individual level, there was lack of training, as well as fears and beliefs, which impacted their capability as well as motivation to perform the bedside ultrasound. At hospital level, issues such as hospital norms, workloads, and staffing in limited resource settings affected their attempts to perform the ultrasound by impacting on their capability, motivation, and opportunity. Looking at the general health system weaknesses, we had issues of relationships and power dynamics between the various subspecialties and cadres of medicine, and the lack of practice guidelines. And this challenged the uptake of bedside ultrasound by the clinicians. So to just look at a few of some of the quotes that we got from the study, issues regarding training were pointed out. As you can see, one of the clinicians said, we need training, especially for the ultrasound. Most of the doctors are not trained in this. Secondly, issues of equipment maintenance, and as you can see in the second quote by another clinician. Thirdly, we had issues of acceptability. These are the power plays between different subspecialties in medicine. Issues of acceptability. Will a junior clinician be able to do a scan which will be acceptable by a more senior clinician? And there were other issues such as time pressures by the clinicians. As you can see from the fourth quote, they say, at times, I don't even want to do the ultrasound because we have a queue to clear. To stop and do an ultrasound will take me forever. If we had more staff, that would help. I know that I can scan, I want to scan, but I have a queue. So I'd rather send this patient to the radiology in the meantime as I sort out the neck. So issues of time pressure from the clinicians. And lastly, we had issues of, would the radiologist take this up and encourage it? This figure just summarizes some of those issues. And if you look at the first column, we have the intangible softwares of fears and beliefs, norms, relationships, and these were looked at from the rows. You can see the individual level, the hospital level, and the system level. And all these are connecting, excuse me, all these connect and affect the motivation, capability, and opportunity of the clinicians in performing the ultrasound, point of care ultrasound. Our results are very similar to those of Sachita and colleagues, and these were done in a resource-limited setting and basically pointed out issues of lack of training, machine malfunction and maintenance, and inability to perform the maintenance on some of these existing machines. In high-volume EDs, like in Africa, we have a lot of, the clinicians have competing demands, lots of patients, limited human and financial resources. And these were quoted as some of the issues as to why the clinicians were not so willing in doing the bedside ultrasound, in adopting it. However, we've had studies done in Malawi, Rwanda, which definitely show viable alternatives to clinician-performed POCUS in limited resource settings. Our study does highlight the need for context-specific ultrasound protocols, which are suited to the needs and available resources in low and middle-income countries. And of course, it showed that training should be embedded within an accreditation model to ensure consistency and upholding of set standards. In conclusion, this study showed that POCUS for the diagnosis of acute breathless patient requires 1, well-maintained and accessible equipment. Secondly, highly trained individuals with time to perform examination who have ongoing support. And this would require appropriate training programs within a framework of supportive supervision nested within existing healthcare pathways. And lastly, strong evidence of benefit. We need evidence to identify the optimal scope of ultrasound examination, the diagnostic benefits, and also the opportunity costs. Thank you. I'd like to thank Impala for sponsoring the research and all my supervisors. Thank you.
Thank you very much. That was fascinating. I'm just wondering, is your work, the outcomes from this, is it going to affect policy in any way?
Yes. This is part of a larger study, which we have a huge quantitative study, which are looking at the benefits and the utility of point of care ultrasound in our setting. And yes, we hope that eventually it will affect policy. And hopefully with the introduction of point of care ultrasound, we can shorten the time it takes for patients to be seen and to be diagnosed.
I'm also wondering, I mean, you mentioned there are limited financial and limited human resources, and I presume there are other issues that are also suffering from the same shortages. Is this the cost-benefit analysis of doing this treatment? Does it supersede others? Is this more vital than perhaps other work that's also suffering from a lack of financial and clinical resources?
That's a very good question. I think we have to identify a soft balance, because the clinicians, we don't have the luxury of having CT scans and the other advanced diagnostics. So patient care is as important. I mean, to us, the main aim of treatment is so that we can save as many patients as possible. So yes, we have competing demands, but perhaps ultrasound offers as a way of basically having better outcomes of patients in a very resource, that doesn't need a lot of money or a lot of human resource, because it can be taught to anyone. It can be taught to a clinician, it can be taught to nurses, it can be taught even to clinical officers. And we could use the task shifting as a platform to train.
Brilliant. Thank you very much. That's the end of this session. I'd just like to ask the presenters to join the stage chat to answer any other questions. Oh, there's one question. One minute, sorry. Apparently it's on the way. Or maybe not. No. It says, okay, it's coming through in bits, I'm sorry. For POCUS, would pulmonary embolism be high in inclusion or exclusion? Or are you thinking more of an ID etiology?
That's a very good question. We actually did a systematic review and looked at use of multi-system POCUS to diagnose pulmonary embolism. We reviewed the literature out there, and it does have a very high sensitivity and specificity, of course, in resource settings that do not have the CTPA, which is the CT pulmonary angio, which is the gold standard for diagnosis. So yes, it can be used to both diagnose and also to rule out PE, and also for ID etiology, definitely.
Great, thank you. So yeah, if the presenters could move into the chat area and just answer any other questions. Thank you to all our presenters. We've had some really interesting and diverse talks. Thank you very much.
Okay, so we're live. Good morning, everyone, and welcome to this third session. We have 5 speakers, and I'd like to remind everyone that these are 10-minute presentations, and we have time for some questions. And I would like the audience to post as many questions as possible, but please do that in the Q&A section, not in the chat, okay? So the first speaker is going to be Beth Bolton from Vector Biology, talking about new genomic tools and rapid automated resistant detection in mosquito larvae. So Beth, I think the stage is yours.
Good morning, everyone. I'm Beth, a PhD student working with Gareth Lycett in the Vector department. Today I'll give an overview of my PhD project, which I'm completing in collaboration with the CETEL group at UCL. Each of my chapters focuses on a different mechanism of insecticide resistance, and I analyse this using several different genetically modified mosquito strains. As many of you will know, despite their size, mosquitoes have a massive impact on millions of people around the world due to their role as a vector in the transmission of disease-causing parasites and viruses. The spread of these diseases can be controlled by killing the mosquito vector. This is usually achieved through targeted exposure of the adult stages using insecticides on long-lasting insecticide-treated bed nets and indoor residual spraying. However, the emergence of highly resistant populations and behavioural changes has reduced the efficacy and effectiveness of these methods. One alternative method we can use is larval control. This involves targeting the aquatic stage of the mosquito by either removing or covering the water sources they live in or treating these sources with insecticides. So the first part of my project is looking at this new larval automated phenotyping assay called NVAP. The reason that this is necessary is because, as with adults, larvae also can develop resistance to insecticides we use to target them. However, unlike adults, the WHO larviciding assay is not great. It involves putting 25 third-in star larvae into fairly large pots of 200 ml of water plus the insecticide and count the number of dead at 24 hours. However, the rearing requirements are quite laborious to get the numbers to test. It's time-consuming to set up. It's very low throughput, just if nothing else, based on the physical space taken up. And there's an investigator bias, which comes where knowing whether something is dead or alive, there are specific parameters, but they can be quite ambiguous, and there's an element of personal determination. So what we have done in collaboration with the team at UCL is looking to develop an invertebrate automated phenotyping platform. So NVAP was developed by the CETEL lab at UCL for analysis of nematode motility, and it's been modified for use with mosquito larvae. So what we do is by filming larvae exposed to chemicals in 96 well plates and examining the variance within pixels over time for each well, a motility index can be calculated which indicates the amount of movement in each well. Simply, the more larvae are moving in a well, the more larvae are alive, because dead larvae don't move. So there's some key benefits of this compared to the assay I described just a moment ago. The rearing time is reduced as first-in star larvae are used. It's high throughput as it can be done in 96 well plates, so you don't have the space issue. The bias is taken away from the data collection as it's a computer algorithm which calculates the motility, not a human person making a yes or no decision. It's very quick to set up. You can use multi-well pipettes, etc. And the physical space required is much reduced compared to the other assay. So I'll just quickly show you some data. So one of the ways that I've been using this assay is to calculate resistance ratios for different strains tested against different insecticides. So to do this, we test a range of concentrations for that strain, build a model, logistical model, to predict the curve for the movement index reduction, look at the IC50, which is the reduction by 50%, and then calculate a resistance ratio compared to comparable susceptible strains. So, for example, I won't go into detail in this because there's a lot of information here, but this is the type of data that you can get out of this in only a couple of months worth of work, which is nice. So I'll move on to the second part of my project, which involves carboxylesterase. It's an alpha carboxylesterase called CCEAE3A, but as it's the only gene I'm talking about here, I'm going to refer to it as the carboxylesterase for ease. Its upregulation in mosquitoes is strongly associated with chemophoss resistance in both field and laboratory selected mosquitoes, and the selection for the upregulation of this gene typically occurs in the larval stages. So, as you can see here, I created a GAL4 UAS system of transgenics in order to upregulate this gene. Putting it simply, the ubiquitin promoter expresses GAL4. When GAL4 binds to UAS, it massively produces the gene downstream, which means that this gene's then it's produced in Anopheles gambiae. I first tested against common larvicides, as this is what's already kind of looked at for this gene, and found resistance as expected to chemophoss, chlorpyrifos, and fenthion. And then I looked at testing adults, because it's very important if we're using larvicides that we may be selecting for resistance in adults as well. It's very interesting here where all the organophosphate adult insecticides there was resistance to, but additionally alpha-cypermethrin caused resistance. I then tested this in partial assays and found a 9-fold resistance ratio, which is quite unexpected and could be very problematic in the field. So my final chapter is what I'm completing at the moment. It's working with a CRISPR-Cas9 line I've used to create ACE1 mutants. ACE1 is the primary target for organophosphate and carbamate insecticides. And using the mosquito homology directed repair mechanism, I introduced the G119S point mutation into a susceptible line. This particular SNP has been strongly linked with organophosphate and carbamate resistance in field samples, but it's never been investigated just in isolation. So when you select mosquitoes in the field or you select mosquitoes in the lab, other changes occur at the same time alongside it, which can be compensatory or can mask the real contribution of a particular change. So it's important to look at what is just that mutation on its own, what impact is that having. And it's important to note that this particular SNP is the only Anopheles gambiae SNP in Anopheles gambiae to have been implicated in resistance for with ACE1. The second part of this project is looking at transcription localization of ACE1. So using this very small F288 protospacer, I'm able to express EYFP in the same place as ACE1 is transcribed. So I don't yet have data gathered for the point mutation line. I'm in the process of doing that. I'm wrapping up that just now, so I'll not present it, but I will show you some pretty pictures of the transcriptional localization. So this is not all the data I have. I've selected 2 of the kind of strongest phenotypes which I have observed, and that is in the dissected thorax of adult mosquitoes, there's very strong expression in the subesophageal ganglion, as expected, and in the third instar larvae, you can observe it in the nerve cord along the dorsal side. I will be doing some confocal microscopy on this to get some really pretty images, but I haven't quite completed that yet. So I'd just like to go through my acknowledgements, particularly my supervisor, Gareth Lysett, and my supervisor at UCL, David Sattel, the two technicians in my group who, throughout my project, have supported me, Annalie and Fraser. And there's many other people, but I don't quite have time to go through everybody just now. Thank you very much.
Thank you, Beth. That was great. We have no questions so far. Please put them in the Q&A. So Beth, you showed some huge error bars in your first story in the imaging for some insecticide or some molecules. Can you why is that?
Part of it is an analysis issue of I'm still tweaking not the analysis of extracting the movement index, but the analysis of the data. So I'm still working on that. Part of the problem we've run into is if there happens to be a zero recording when the mosquitoes are actually alive, which occurs rarely. But in some of them, we had only been doing 5 kind of recordings per time point, and it turned out that wasn't quite enough. So we started taking 10, and that did solve that problem a little bit. And it's reduced the error bar slightly. Does that make sense?
Yeah, I guess. So we have another question. Someone says, lovely pictures, Beth. Are the heterozygous fluorescent throughout, just weaker than the homozygous, or are they only fluorescent in the tail regions, as the picture seems to show?
So they are a lot less brightly fluorescent than the homozygotes. There is some fluorescence in the head, but it's a lot less strong than the homozygotes. There's a very, very large difference. Because the homozygotes are so bright, in the images taking them together, it's quite difficult to represent just how, that the heterozygotes are also quite obviously bright as well. But yeah, there is some fluorescence in the head, but it's much stronger in the tail in the heterozygotes.
Okay, thank you, Beth. I think that's it. So we're moving to the next speaker, who is Brenda Mungai from Clinical Sciences, with a talk titled, If Not TB, What Could It Be? Chest X-ray Findings from the 2016 Kenya Tuberculosis Prevalence Survey. Are you ready, Brenda? Yes? Okay, all yours. Brenda, you are muted. Can you please unmute?
Sorry about that. So, good afternoon and good morning, some of you. So my name is Brenda Mungai, final year PhD student in global health. So I'm going to present about abnormalities other than TB found during the chest X-ray prevalence survey 2016. That is the outline of my presentation. So TB is still a big problem there, and we still have 3 million people with TB diagnosed and underreported. And this was before COVID. These numbers have increased drastically because of underreporting and underdiagnosis. We know that chest X-ray is an important tool for TB screening and triaging. And with new guidelines, but also recently chest X-ray machines with low operational costs that are easier to use, and new technology and innovations easier to use, and portable X-ray machines, then use of chest X-ray in lower middle income countries for screening will be more likely. Now, we found that when people concentrate on TB screening, there's you mostly want to see whether the abnormality is related to TB, and if there are, then you go on to bacteriological confirmation, and therein is a gap as to quantifying those abnormal other than TB. What problems do they have? And since this represents a missed opportunity for identification and treatment of potentially significant disease. So the aim of my research, therefore, was to quantify and characterize non-TB problems identified by a TB-focused chest X-ray screening during the 2016 Kenya Prevalence Survey. And so the findings of this study then informed a modeling projection to estimate the range and prevalence of lung conditions and what service provision need to be in place if chest X-ray adopts TB if Kenya adopts chest X-ray screening for TB. So I'll give a brief overview of the prevalence survey because that's where my study population came from. So for the prevalence survey, there were 63,050 enrolled participants who underwent 1 symptom screening using cough of more than 2 weeks, and 2 chest X-ray was offered in parallel. So I will concentrate mostly on those that received the chest X-ray screening, who are 62,484. And we can see from those that got chest X-ray, 81% had normal findings, 10% had abnormal suggestive TB, and 8% had other abnormalities. And so for this study, my study population was actually those with abnormal suggestive of TB and abnormal other. So the images that had been classified as that. The digital images came with some accompanying clinical information. Since from literature, we did not have the prevalence of chest X-ray pathology in a population-based screening, did a pilot study that was between 2018 December and January, and engaged 5 radiologists who did single reading of films, and we targeted to have 500 films as our sample. We developed a reporting tool. We adopted some from the prevalence survey and modified, and then we trained the radiologists on this, and also we had some chest X-rays to just ensure that the understanding of the tool and interpretation of the diagnosis that we had listed out. And this is a screenshot of what the radiologists were feeling on their end, looking at primary diagnosis, differential diagnosis within the major categories. So the pilot results, most abnormalities were heart and great vessel at 36.6%, with lung abnormalities at about 24%, and the others as shown. In the abnormal other category, cardiomegaly was actually the highest prevalent condition, and so that was at 35.5%, with some other lung abnormalities. In the abnormal suggestive of TB, old latent TB was what was the highest prevalent at 13.9%. So we used these findings to then calculate the main study sampling size to 3.5% of true value with 95% confidence interval, and we came up with a sample size of 1,140 films, and we conducted this study in May and to July 2019. 10 radiologists were involved. So the images were reviewed by the radiologists, blended to the field classification, but also to other study radiologist reads. And then for any discrepancies, we had a third radiologist to resolve this, and that's what we use for the analysis. Now, as part of the results, so the median age of our sample was 51 years. Among the 600 abnormal images, 21% had multiple abnormalities, but the highest abnormality was actually cardiomegaly, and then also mild, moderate post-TB lung changes. So cardiac and pulmonary diseases accounted for 66% of the non-TB abnormalities, and the breakdown was cardiomegaly in 23%, non-specific patterns 4.6%, some suspected COPD, chronic obstructive pulmonary disease at 3.2%, post-TB lung changes at about 7.6%, and for some we found some mediastinal masses. So the overall agreement between readers was moderate at 0.41, and I had said about the tie break read by third radiologist who had the knowledge of the first two radiologist read. So what's the implication of my findings is that more and more countries are embarking on chest X-ray TB screening activities, and they now need to know that other treatable and preventable lung conditions may actually be as likely or even more prevalent than active TB. And so it's good this can aid in early detection of preventable and treatable chronic lung diseases. However, through the health system, we also need maybe to integrate NCD screening in mass screening activities, because cardiomegaly in our setup could actually be due to complications of non-communicable diseases. We also need to set up structured referral mechanisms and NCD prevention and messaging. Part of what we've done with these findings is had policy engagement during the Kenya Scientific Lung Conference, and then also feedback to the National TB Program during part of the modeling forums, and we've also published these findings. Thank you very much. This was made possible by the Impala funding through NAHR.
Thank you, Brenda. So we have one question that says, are your patients coming from a schistosomiasis endemic area, as some of the other might be pulmonary schistosomiasis? Thank you.
Okay, so I think that's a good question. In some of the counties, yes, there's actually schistosomiasis. And yes, that could actually be a possibility. As you may note, we were just using the radiology diagnosis. So at clinical level, they also need to probably investigate those as part of the interstitial diseases that you could find.
Brenda, I'm wondering if you are expecting perhaps COVID having an impact in this kind of service in the future.
Yes. So yes, one. So you can look at it both ways. So there's one positive, because with chest X-rays out there, and knowing that part of what has been developed as the computer-aided detection technologies, there are some with actually for COVID and for TB. So we could actually be combining those during the screening activities. I think there's going to be more focus on finding more TB patients now that I've said COVID has really affected case finding in the year 2020. And so maybe scale up of this due to the COVID pandemic.
Okay. We have time for another question, I think. How does agreement between readers' value, which is quite low, compare with other studies examining these or similar types of chest image data?
Okay. Yeah, so I think radiology is one of those that, unfortunately, agreement between readers is pretty low in most cases. I think moderate. I think there's one study that we looked at, just to note that this specific one of looking at X-rays during a mass screening activity, there was no contemporaneous evidence, and so part of it were earlier studies in Europe. But yes, moderate agreement is actually maybe what you'll find. It varies a lot. I think radiology intra-reader agreements are quite varied. Inter-reader.
Okay, so thanks very much, Brenda. And we are moving to the next speaker, who is Joshua Longbottom from Vector Biology. His talk is optimizing passive surveillance of neglected tropical disease in the area of elimination, a model study. So Josh, we are ready when you are.
Great. Thank you, Aitor. So hello and welcome to my presentation. I'm Joshua Longbottom, a third year PhD student working with Michelle Stanton and Steve Torr. So today I'm going to be presenting on one of my PhD chapters. So the work I'm presenting has been published in PLOS NTD, but unfortunately due to the breadth of the work and the 10-minute limitation for the talk, I'm going to have to cut out quite a lot of the juicy bits of the methods, but all the details can be found in these two papers here. So the paper on the left mostly goes into the construction of the friction surfaces, which are vital for this analysis. I'll describe them briefly. And then the paper on the right discusses the whole process with regards to optimizing disease surveillance. So I'll start with a bit of background on disease surveillance. Surveillance systems are essential and they're usually seen as the first defense for detecting emerging infections and are necessary for endemic pathogens. When a disease approaches elimination, the number of cases decline and treatment costs diminish. However, surveillance costs remain high even after the last case. So some good examples of this are polio and guinea worm, where there's intensive surveillance to detect the remaining cases. So frequently evaluating surveillance systems ensures that they remain operating at maximum efficiency and theoretically at the optimum cost. However, there's a lack of guidance on how best to do this. And if this scale back isn't performed considering a range of different factors, it could result in the underreporting of disease, which is not what you want. So I hypothesize that that surveillance systems can be optimized by carefully considering the spatial placement of sites. And for this work, I'll be demonstrating a methodology applied to Gambiense human African trypanosomiasis, known as sleeping sickness in Uganda. So Uganda is on track for GHAT elimination, which is defined as fewer than one reported case per 10,000 people per year across all health districts. So ramping up of surveillance was performed in 2015, increasing the number of surveillance facilities from 3 to 170. However, as we're now seeing a decrease in the number of cases, there's a need for scale back. So the previous scale up, sorry, there's a lot of mismatch of words here, but previous scale up used the Euclidean distance, so straight line distance, as a measure of accessibility, so to see how far individuals live from a surveillance center. So that could be represented in kilometers, for example. But we know that other factors influence how accessible a location is. So whether or not there's a road present, the type of terrain, geographic barriers, available transport, for example. So there's ways to improve upon this approach by considering additional estimates of travel time, particularly incorporating travel time. So whilst estimating coverage in general is useful, it's also beneficial to have coverage goals. So during a meeting with WHO in April of 2019, new goals were proposed for the 2030 roadmap. So one of the goals for GHAT was to ensure that 50% of the at-risk population lived less than 1 hour's travel from a diagnostic facility, and that 95% of the population lived less than 5 hours travel. So this target posed two research questions, which I hope to address during my PhD. So what number of facilities would be required to meet these targets, so the 50% coverage and the 95% coverage? And where should the optimal number of facilities be placed? So once I've derived that number, is a way that they can be spatially arranged, which results in the maximum benefit. So unfortunately, this target wasn't ratified, so these coverage values. However, they do form the basis of my work. Seems to be a good benchmark for quantifying surveillance coverage. So to address these questions, I aim to use what's called a cost distance analysis. And for this, you produce what's called a resistance surface.
And this is a gridded surface, so standardized grid cells, where the time taken to cross each grid cell is defined. So in this image here, each grid cell represents 30 meters by 30 meters on the surface. And the values represent how easy or how quick you can travel through a certain location. So roads would be associated with a lower cost, so a faster travel speed, and dense vegetation or off-road would be associated with a higher cost. So using an origin and a destination, you can compute what's called the least cost path, so the most cost efficient or optimal path across this resistance surface. And just using either one or several destinations, you can produce what's called a cost distance surface. So this is a cumulative cost. So within each 30 meter by 30 meter cell, it's a total cost to travel from that cell to the origin or the destination, which is marked here as A. So when you have this surface, so this cost distance surface, you can combine it with estimates of population coverage to quantify the size of the population living within certain distances or travel times. So I'm going to use a cost distance surface to address question 1, which was the number of surveillance sites. So this can be answered using a simulation-based approach. So I have 170 facilities available. What I'll do here is I'll use one facility, calculate the percentage coverage, and increase that to 2 facilities, then 3, for example. And I'll keep going until I see a plateau in the coverage or until I hit about 50% and 95% target. But the obvious issue here is as you increase the number of facilities, the number of available combinations increases up until roughly 85 facilities. So it's highly computational. These are just some images to show how I would construct a surface with one facility and then with 2. So what I opted to do to kind of reduce the computational constraints was to choose a random sample of 1,000 facilities for each number of n, where n was from 2 to 170. So I'll generate different spatial samples, and then using those, I'll incorporate them with the cost distance, the resistance surface, and generate a cost distance output, and then quantify the population. So I'll have 1,000 random combinations of, for example, 120 facilities, generate the outputs, and then quantify the coverage. And then using that, I can look at an average coverage for a select number or whether or not any out of the 1,000 hit the target. So some results. Surprisingly, a very low number of facilities are required to meet both the targets. So this could be achieved by using just 3 facilities, which is a massive scale back from 170. Perhaps it's not feasible in this operational sense. So for the second question, the spatial placement, we decided to use a predefined number, which was 54 facilities. So this number arose from discussion with health district officials from another project, Japan. So to compare the coverage surfaces, I generated estimates of travel time using all 170 facilities. So the figure on the left is the continual travel time, and then it's categorized in 30-minute bins on the right here. And I show that for all 170 facilities, coverage is quite high. So 96.15% are within 1 hour, and 99.7% are within 5 hours. And using the 54 facilities, I can hit a similar level of coverage if I optimize the spatial placement. So I go from 96.15 to 95.12 within that 1 hour travel time, which is quite good. So using the 2 surfaces, I can kind of show where the spatial differences are, and so what locations would be most affected by scaling back. But how the algorithm works, because it considers population distribution, these tend to be locations which are not densely populated. And so to summarize, there's a need for technical guidance on how to monitor distribution and incidence of infections. I show using this optimization approach that the number of facilities could be reduced from 170 to 54, so 70% reduction, without any effects on any significant effects on population access. So these simulations can be repeated as the spatial distribution changes over time. There's other factors to consider, economics, politics, and how feasible this is from the operational sense. But as a methodology, this is quite an interesting finding. So yeah, I'm sorry I'm rushed for time. I'd just like to thank and acknowledge my supervisors and funders and collaborators for their help and support during this project. Thank you.
Thank you, Josh, right in time. So a question says, East Nile region has fewer cases than West Nile region, so cross-border pathways from DRC and South Sudan are important. If this change, could your sentinels need resiting?
Yes, they could. I don't think the current surfaces I have would be able to address that question because I've just created the friction surface for Uganda. But it's easy enough to expand to cover, kind of bordering areas of DRC and South Sudan. But yeah, as the population at risk changes, I can just feed that new surface into the same algorithm and derive, a new optimum placement or number. Yeah, good question.
I do have a question. I'm curious to know if you or your team have been able so far to, put your models and your predictions to good use, and you've been able to see the, a real outcome from your work.
Yeah, so once I generated the results from the scale back piece of what I've just presented here, we were in touch with members at WHO about the results. One concern they had was that the population at risk surface I used was different to what they were using, but were very keen on the methodology. And one thing they stated was if they provide the new risk surface, could I repeat the analysis and give them the results? So that's something I do plan to do, but I've just moved on to the next chapter of my PhD at the moment. But yeah, there is interest in what I'm doing, but yeah, validating these kind of things is quite difficult as well.
Okay, that's really good. So that's it, Josh. We are moving to the next speaker, who is Martin Jorogi from Clinical Sciences, talking about changing lung function and associated health-related quality of life, a 5-year cohort study of Malawian adults. Are you ready, Martin? We can't hear you. Not yet. No. Maybe something with your mic.
Hi, Aito, are you able to hear me?
Now, yes. Go ahead.
All right. Thank you for that. So let me start. So good afternoon, everyone. I am Martin, a third-year health economics PhD student working with Professors Graham Devereux and Louis Nielsen. I will give a presentation, as mentioned before, titled Changing Lung Function and Associated Health-Related Quality of Life, a 5-year cohort study of Malawian adults. This study has resulted from a survey that I have been leading in the past 3 years. Chronic obstructive pulmonary disease and asthma are the most prevalent chronic respiratory diseases globally. COPD accounts for about 3 million deaths annually, 90% of which occur in low-middle income countries. In Malawi specifically, previous epidemiological bold studies have documented a large proportion of atypical lung function of about 8% and 32% obstruction and restriction in rural Malawi, and 4% and 39% of obstruction and restriction in urban Malawi. These few studies have documented a prevalence of atypical studies. However, the economic cost and the health-related quality of life of the adults has not been documented. It is important, and this is important because the risk factors for CRDs are such as lung infections and environmental factors and material deprivation are usually potentially modifiable. So due to these circumstances, my PhD work has 3 main research questions. What is the estimated health burden of airflow obstruction in Malawi? Second, what is the estimated current and future economic burden from a societal perspective? And finally, what are the estimated cost effectiveness of selected key interventions for adults with airflow obstruction and related scale-up costs? In order to address my research questions, we conducted this study with the aim of first, estimating the annualized rate of change of lung function by repeating spirometry, and secondly, establishing the health-related quality of life outcomes associated with the changing lung functions. Onwards to the methods. We followed up a cohort of adults in Chikwa, Malawi. Chikwa is located in south of Malawi, and it's a predominantly rural area of Malawi. The outcomes of interest that we're interested in for the lung functions were the forced expiratory volume at 1 second and the forced vital capacity, which were then compared with the Global Lung Function Initiative and derived Z-scores to kind of be able to use them usefully. And other secondary outcomes are the health-related quality of life utility weights, which we obtained through the use of the short form 12 questionnaire and the veterans 12 questionnaires using the short form 6D algorithm. We used the t-test and the chi-square test for the univariate analysis and simple linear regression methods in the bivariate analysis to assess important covariates independently. These results are useful in building a mixed effects models where random effects were accounted for at the individual level and fixed effects were selected sequentially using deviance testing and likelihood ratio testing and a maximum likelihood estimation. In our results, we're able to collect data from 1,232 participants, which is about 83% of the baseline, of whom 1,082 provided acceptable spirometry, about 73% of the baseline. The follow-up participants were older, had less years of education to the baseline participants. No other significant demographic characteristics are found between the follow-up and the baseline. Compared to the baseline, the 249 participants from whom we are not able to follow up were older, they had less years of education, and had lower health-related quality of life scores. Looking at the ventilatory functions, we classified the spirometry readings into patterns consistent with clinical diagnosis of COPD, asthma, restriction, and normal lung function. This then becomes useful to clinicians to kind of make sense of the results that you're presenting. We found that while the prevalence of those with asthma and restrictive lung disease remains constant, those with patterns consistent with COPD increased, while those with patterns consistent with normal lung function decreased. In the mixed effects modeling, forced expiratory volume declined by 53 milliliters per year, and forced vital capacity declined by 45 milliliters per year. The significant associated covariates were participant age, their height, their body mass index, their sex, and previous TB diagnosis. These associations remained constant even when the forced vital capacity scores were modeled as Z-scores, as outcome variables. Onwards to the health-related quality of life. There was an obvious challenge with the health-related quality of life because this data was not collected in the 2016 and 2017 follow-up. It was only collected in the baseline at 2014 and in this follow-up at 2019. And looking at them, there was a decline of about 0.096 in the follow-up compared to the baseline. And this decline was seen across all lung functions and those who had reported previous diagnosis of TB. In the bivariate associations, compared to those with normal lung function, the health-related quality of life of those with asthmatic lung function declined by 0.08, those with restrictive lung function declined by 0.05, and those with COPD declined by 0.04, while those with diagnosis of TB declined by 0.09. The results from the mixed effects models, the health-related quality of life associated with the follow-up time, older age at baseline, and female sex, but it was not affected by years of schooling or smoking. The symptoms of shortness of breath, wheeze, and cough were also independently associated with health-related quality of life. It was also important to note the health-related quality of life was associated with previous TB diagnosis and forced expiratory volume, but not the forced vital capacity in the multivariable models. So, some key findings. First of all, forced vital capacity, FEV1 declines faster than forced vital capacity. Second, from the spirometric patterns, comparing the baseline and this follow-up, there are significant differences in those who are found to have COPD and normal lung function, but not those with asthma and restrictions, suggesting no new restrictive cases or increased asthma. Previous TB has a significant effect on the decline of lung function. The key findings related to the health-related quality of life, there was an overall decline in the health-related quality of life, but those with asthmatic patterns had the largest decline, suggesting an exacerbation effect. And previous TB diagnosis has an effect on the decline of health-related quality of life. Health-related quality of life is associated with symptoms and abnormal lung function. In conclusion, first, the lung function decline has an effect on the quality of life. Second, COPD is common in this population, but because of suboptimal lung growth and lung function loss due to TB diagnosis. This is important because it has implications on the design of interventions that could then be recommended. Third, the health-related quality of life is adversely associated with symptoms and abnormal lung function. Fourth, the health-related quality of life differences exceed the minimally important differences for the SF6D tool. The minimally important differences are when you administer a tool, the difference that would then be noticed that would then be clinically important to intervene. And finally, early intervention could help in the slowing down the decline of lung function and thus lead to improved quality of life. I would like to acknowledge and thank the people on this slide who participated in this study and the funders, specifically Impala through NHR. Thank you.
Thank you, Martin. It's time now for questions. So we don't have questions so far. So Martin, if you did these studies in, let's say, a different region or a different country, would you expect similar results? Meaning, how variable are all these?
So that's a very good question. So we would expect similar results. However, the rate of decline that we found was quite high, comparable to other regions. So that decline of about 53 milliliters per year of lung function is equivalent to about a person who smokes about 15 cigarettes in a high-income setting. And that shows that then the challenge is actually then that the intervention is not getting there early, which means the rate of lung function is quite quick, leading to then mortality, earlier mortality and lower health-related quality of life because of just that lack of intervention. So it's that's what you'd expect. And yeah, I hope that answers part of the question.
Yeah. There's another question. It says, thank you for a really excellent presentation, Martin. You have important findings. What interactions have you had with policymakers who might take action on them?
So part of this work was kind of me talking with the policy developers, specifically the Ministry of Health officials, to kind of help that, because this is the first part of a broader study that is aiming to design better interventions for scale up, especially for low resource settings such as Malawi. And that includes a lot of conversations with the Ministry of Health policymakers, so that we can then recommend the most cost-effective interventions to scale up at the most affordable price. And it has, so far the feedback has been great, and it has been good trying to network with them and contact them.
Okay, thank you, Martin. That's great.
Thank
you. So we are finally moving to the last speaker, Irene Ayakaka from International Public Health, talking about communication for chronic lung disease, insights from people seeking care at the 3 hospitals in Kampala, Uganda. Are you ready, Irene?
Yes, I am. Can you hear me?
Yes, all good.
Oh, all right, thank you. I've been introduced, Irene Ayakaka from the Impala Lung Health Program at LSTM. I'm a PhD student in my 3rd year, and my presentation is on one of my chapters, which is communication for chronic respiratory disease management, insights from narratives of people seeking care in 3 facilities in Kampala, Uganda. As way of a background, about 550 million people were estimated to have CRD, that is from the Global Burden of Disease study in 2017. Many of them live in low and middle income countries where premature mortality due to CRD is highest and where there are less resourced health systems. So chronic respiratory diseases or CRDs disrupt people's lives greatly and interfere with activities of daily living. The medical management of CRD is greatly augmented by self-management strategies. And in fact, it is an essential adjunct to medical management. But for self-management, there's a need to guide people seeking care or people who have a diagnosis of CRD to enable them to live with CRDs and their disruptions and to manage these disruptions. There is a need for patient-centered approaches, including patient-centered communication to guide patients and families to appreciate and utilize information. And finally, a need for effective communication for this to happen. In this low-income setting, little is understood about the modalities of providing patient-centered communication for chronic care, in this case for chronic respiratory disease care. The health system is mainly tailored to manage acute communicable diseases, and as such, chronic diseases are often managed as serial acute episodes. So this was a qualitative study to describe people's experiences related to communication as they sought for health care for CRD and symptoms of CRD in Kampala, Uganda. The next 3 slides really show, they illustrate the context within which this study was carried out. A busy health center 4 in downtown Kampala. It is an outpatient setting and sees over 500 patients every day. It serves a really overpopulated area with immigrants, refugees, and low-income earners, with over 750 people notified for TB every year. We have a national referral hospital which provides specialized services, including inpatient services and a specialized chest clinic. And as a national referral, it receives patients from all over the country. And the 3rd one was a specialized lung disease institute or unit. Our data collection methods involve longitudinal patient narrative interviews guided by topic guide with open-ended questions. We had FGDs with a topic guide with open-ended questions. We're exploring participant perceptions of communication, but also their experiences. And we did a direct observation that was guided by a checklist. We observed at various points, we observed the registration, the screening, the waiting areas, but also the clinical consultation, the dispensing, and the kind of documentation that was happening. For data analysis, The discussions were recorded and transcribed for analysis, and many of them were conducted in the local dialect. But where this was the case, we transcribed and translated. We translated into English and transcribed. We used in vivo for inductive analysis. All transcripts were. We used open coding to identify initial emerging codes that were related to communication. And an initial code book was updated as we got new codes, and updated iteratively and discussed and refined with the help of the PhD supervisors. So we observed the general flow of patients, healthcare worker consultations, as said, and held 14 longitudinal narratives at 3 different time points. Starting in June 2019, we had started with focus group discussions. However, when we shut down or locked down the country due to the COVID-19 pandemic, we did not continue with the FGDs. We interviewed so far 10 health workers, but the interviews with the health workers are not included in this presentation. So what were our findings? 3 major themes emerged as follows. 1, communication was key, not only to understand illness symptoms and treatment, but it enabled people seeking care to navigate communication within their families, but also to make sense of altered relationships as a consequence of the disease. Communication played a major role in shaping health-seeking behavior and navigation within health facilities and between health facilities. And thirdly, communication, the key model or the predominant model used here was provider-centered, and this was across both the public and private facilities. So, in going in depth with looking at the 3 themes, people sought to seek understanding of symptoms to make sense of an altered life. A life that was altered by CRD and or symptoms related to CRD. An example is Ibrahim, a 47-year-old male who needed to understand how to continue looking after his family, and yet his health had been greatly impacted by CRD. People seeking for treatment for CRD did so across a fragmented system. In context without linkages between the formal and informal, between the private and public health systems, as the case in Uganda, it became very challenging. Eunice, a 61-year-old woman, experienced this as she had a medical emergency one night and decided to go to a clinic. At the clinic, she says, I tried to explain to them that the medicine had overpowered my body. I wasn't feeling well. I tried much to explain to me that there are some types of medicine I can't use. It appears like it has some salbutamol in it, but they gave it to me and I felt so bad. I started shaking so much and felt like my heart was moving out of my chest. So the linkage between private and public facilities is extremely poor. In navigating the health system, both private and public facilities at different tiers of the health system can be accessed by patients, and this is usually by initial preference, ability to pay for care, and other factors like availability of health facilities in the vicinity. Patients seeking CRD relied on health workers to refer them to the right facilities for appropriate care. According to the narratives and the FGDs, health workers at private facilities often failed to refer patients even when they were unable to manage them. And the major motivation for this was the money that they get from the patients. Second, patients or people seeking care faced very many challenges navigating within health facilities. Even for those with the ability to read and write, some challenges like negotiating for free care when they were entitled to it failed. A participant from the focus group discussions said that even as you move along the corridors, because I'm a bit educated, I can read some words. They say, don't pay anything. Why then should a health worker say pay? Why do you have to get something out of your pocket first? And yet this is a government hospital. Finally, the provider-centered model, it did not set a conducive tone for patient-centered communication. Most times, patients or people seeking care complained that they were unable to seek for clarity or ask questions of health providers, and coupled with their expectation of their role and the role of the health workers in communication, it highlighted poor relations between patients and health providers. Communication was majorly unidirectional, very brief, often took the form of questions and replies from the people seeking care. And patients or people seeking care did not appreciate that the health workers seemed intent on just prescribing medications without explanation. And the provider-centered model was actually a barrier to gaining understanding. The time spent between patients and people seeking, between health workers and people seeking care was too short to facilitate meaningful discussions. All I know is that my lungs are sick. I can't do any work. I can't do most of the work, but I was not told when and if it would heal. So apart from coming back whenever I am told to. In summary, the nature of communication between patients and providers often led to incomplete understanding of illness and the chronic nature of illness, as well as occasionally dangerous episodes. In addition, patients articulated frustration with their experience of disjointed and fragmented care, pointing to a need for a more coherent approach to communicating different aspects of CRD management. They expressed a desire to have health workers communicate the diagnosis in a way that enabled them to understand symptoms and illnesses. So the need for a more coherent approach has led more recently to the person-centered model being suggested. It theorizes a three-step process where you build rapport between the patients and providers, you share information enhanced by strong rapport and the relationship built, and finally, shared decision-making. However, in our context, the challenge remains how to operationalize this model in a context of fragmented health systems with competing priorities and an overburden of infectious diseases and no chronic care models in place. This is an illustration got from Tran 2020, showing the patient-centered communication model that has been suggested. Study had limitations. The qualitative nature did not facilitate quantification of perceptions and practices. Used only facilities in Kampala. However, there were 3 very high volume facilities at different tiers of the health system, which increases the possibility for generalizing some of the findings. The strengths were the longitudinal narrative interviews before and through the COVID-19 pandemic, and it provided a way to triangulate some of the findings. In light of the paucity of data about communication in this setting, the study provides some insights that can be drawn on the successful interventions to improve communication and patient care. We thank the NIHR Global Health Unit through the Impala for making this work possible. My supervisors, and above all, the patients and participants in this study. Thank you very much.
Thank you, Irene. We have time for one question only. It says, well done with your patients. Did you also see those living with HIV and a long-term ARV having chronic lung disease?
Yes, most of the patients had a history of tuberculosis. Many of them actually in chronic care for HIV. And another finding is many of the patients actually had multiple complaints, multiple illnesses, and some of them had multiple diagnoses. So quite a complex bunch of patients.
Thank you. So we need to finish here. That's the end of the session. So I'd like to thank, first of all the speakers for fantastic presentations, the audience, and also the technical staff, who's been great in the backstage. I just want to remind everyone to attend all the sessions, poster sessions, networking sessions, and maybe keep up with the discussions, science discussions in the Q&A. Thank you very much, and see you all later.